Görebilir misiniz? İnternette mi bir problem var acaba? Çünkü ne mail geldi ne de link.
Cank'a düşüyor olabilir Mehmet. Bir oraya bir bak istersen.
Efendim?
Cank'a düşüyor olabilir. Cank mailini.
Şu an YouTube link yayını başladı hocam.
Peki. Tamamdır. Gelmiş.
Başlayabilirim isterseniz.
Tam o linke tıkladım. Şimdi bir daha gönderebilirsiniz zannedersem.
Problem çözüldü mü?
Galiba evet.
Başlayabilir miyim?
Tabii.
Sayın Rektörüm, Sayın Bölüm Başkanım, kıymetli hocalarım ve kıymetli katılımcılar, bu yıl 9.sunu düzenlediğimiz bölümümüz için bir gelenek haline gelmiş olan Biyomühendislik Günleri'ne hepiniz hoş geldiniz. Bu yılda sizlerle birlikte olmanın heyecanı içerisindeyiz. Bu yılki organizasyonumuzun bir istikrar ve özverimli olduğunu belirtmek isterim. Bu sebeple başta bölüm başkanımız Doktor Tuğba Sevimoğlu'na ve kulüp yönetiminde görev alan, bu etkinliği planlarken görev alan kulüp başkanımız Mehmet Akyıldız, Bahar Gezer, Ümmühan Akbal, Gizem Kanat, Naile Emel Agun ve Nasman Jini'ye teşekkürlerimi iletmek isterim. Davetlerimizi kabul edip bizleri onurlandıran değerli hocalarımıza, bölümümüz ve arkadaşlarımız adına teşekkür etmek istiyorum. Ayrıca uluslararası öğrenci arkadaşlarımız için de kısa bir İngilizce giriş yapıp daha sonra sözü Tuğba Hocam'a bırakacağım. I will continue in English, dear friends. Dear Doctor, Head of Department, and dear professors and esteemed participants, welcome to the 9th Bioengineering Days, which has become a tradition for our department since the establishment of the university. We are really excited to be with you this year as well. So I would like to state that this organization which we organized this year is a product of stability and devotion. I would like to thank to Head of Department Dr. Tuğba Sevimoğlu and especially the club management, to my friends, especially Chair of Club Mehmet Akyıldız, Bahar Gezer, Ümmühan Akbal, Gizem Kanat, Nasman Anjirini, and Naile Emel Agun. And the team that took charge. On behalf of our department and friends, I would like to thank two precious teachers who kindly accepted our invitation. I'm giving the floor to department chair without taking much your time. Please, Dr. Sevimoğlu, we are listening to you. Thank you.
Teşekkürler Şeyma. Thank you Şeyma. Ben de Şeyma gibi önce bir Türkçe giriş konuşması yapıp onu biraz da İngilizcesini çevirmiş olayım. Hepinize hoş geldiniz diyorum 9. Biyomühendislik Günlerine. Öğrencilerimize hoş geldiniz diyorum, katılımcılarımıza hoş geldiniz diyorum. Bu yıl Şeyma'nın da bahsettiği gibi 9.sunu düzenliyoruz. Üniversitemizin kuruluşundan itibaren olan herhalde tek etkinlik budur diye biliyorum. Biyomühendislik bölümü de zaten üniversitemizin kuruluşundan beri var olan bir bölüm. Bu yıl Allah nasip ederse 5. mezunlarımızı vereceğiz. Ben sanırım 3. mezunlardan beri üniversitedeyim. Bu Biyomühendislik Günleri tamamen öğrencilerimizin düzenlediği bir etkinlik aslında. Ben de onların hani kulüp danışmanıyım ama bütün görevleri öğrencilerimiz yapıyor sağ olsun. Gerçekten büyük bir yükün altına giriyorlar. Özellikle burada kulüp başkanımız Mehmet, ve Başkan Yardımcımız Şeyma'ya çok teşekkür ediyorum. Her işin peşinden koşuyorlar. Tabii kurumsal iletişime de buradan çok teşekkür ediyorum. Sürekli olarak maillerimizle onları darladık ama her türlü yardımı yaptılar. Sağ olsunlar, yazılım ekibimize çok teşekkür ediyorum. Şimdi bu program normalde tabii ki yüz yüze olduğu zamanlarda, bu da acıklı bir şey tabii, eskiden bahsediyoruz. Daha şeydi, hani nasıl diyeyim, bizim için daha rahattı. Hani biz oturuyorduk, konuşmacılar geliyordu, konuşuyorlardı. Arada bir öğle yemeği yiyorduk. Bir kaynaşma söz konusu oluyordu. Şimdi böyle uzaktan olunca daha böyle enteresan bir hal aldı. Yani işte konuşmacılara e-mail'le ulaşıyor öğrencilerimiz. Karşı taraf da sağ olsun kabul ediyor. Yani iki taraf da birbirini görmüyor aslında. Bir güven üzerine oluşmuş bir ilişki, çok çok değişik. Geçen sene biz bu etkinliği Mayıs'ın birinci haftası Cuma günü yapıyoruz. Her zaman, yani kuruluşundan beri o şekilde. Geçen sene tabii pandeminin üçüncü ayına mı denk geldi? Mart'ta başladı, Mart, Nisan, Mayıs. O sırada bütün etkinlikler iptal oluyordu. Biz, yani yüz yüze olması gereken bir etkinlik sonuçta. Biz o etkinliği hızlıca bir şekilde, yine kurumsal iletişimle, yine üniversitemizin tüm birimleriyle hızlı bir şekilde online'a çevirdik. Yine güzel bir etkinlik gerçekleştirdik. Bu açıdan şimdi bu sene biraz daha aslında rahatız. Yani daha böyle kolay ilerledik gibi. Önümüzdeki seneler için bilmiyorum artık nasıl bir program izlenir, ama sanki birazcık da böyle hem yüz yüze hem online olması daha iyi olur. Yani şehir dışından da katılımcılarımız olmaya başladı. O açıdan belki online olması daha iyi olabilir diye düşünüyorum. Ben şimdiden takip eden herkese, konuşmacılarımıza çok teşekkür ediyorum. Biraz da İngilizce bir konuşma yapıp Nevzat hocamıza sözü bırakacağım. Sağ olsun kendisi bu etkinliğin başından beri destekliyor. Her sene bizim açılış konuşmamızı yapıyor. O açıdan kendisine de minnettarız. Okay, so let me let me talk a little bit in English. So I would like to thank our students and our speakers for being here in 9th Bioengineering Days. This event is an event that has been taking place since the establishment of the university. So it's that's why it's the 9th Bioengineering Days this year. And this is actually a students' congress, and everything that is related to this congress is actually done by our students, bioengineering students. I'm sorry. So firstly, I would like to thank our bioengineering club management, Mehmet Akyıldız, Şeyma, and I can't remember all the names, but Beyan, Nasma, Bahar. So I would like to thank you guys for preparing this event. It's a big event for our students, and these students are mostly third year and fourth year students. So I'm hoping we'll have many more bioengineering days, and these students will actually graduate and become speakers for us. So I'm hoping that we will see those days coming. This year we're gonna have our fifth graduates, I believe. So because bioengineering department is a department that has been here since the establishment of the university. So in our faculty, there are 9 departments. In the engineering and natural sciences department, there are 9 faculty, there are 9 departments, and some of the departments are new, so they do not have any graduates, but we as bioengineering department have a lot of graduates right now. So and I'm hoping that we'll have many more graduates, and they'll do really nice things for the world. So I'm saying lots of students in our attendees, and I want our students to ask questions to the speakers. So please get ready with your questions, and hopefully our speakers will answer all your questions. I would like to thank the speakers also for accepting our accepting to be a speaker in our Congress. So yeah, thank you all for being here, for supporting us, and for trying to learn new things. We're gonna have different topics with each speaker, so we'll be learning lots of new things actually, and this is gonna be good for all of our students and the participants who are participating from our universities, or maybe I think some labs, and even outside Turkey. I think there are some participants, there are some attendees. So
From Iran.
Sorry?
From Iran.
From from Iran. So maybe next year we'll do a more international program. Maybe we'll have more speakers attending from other countries. We'll see about that. So inshallah, everybody will enjoy this program today, and I thank you all again for joining us.
Thank you, Doctor, for your speech. I think we can wait 5 minutes for Professor Dr. Nevzat Tarhan, isn't it?
Okay.
Nevzat hocamızla bir bağlantı var mı şu an? Hocam görüşüyor musunuz?
35 geçe girecek herhalde.
Peki 2 dakikamız var.
Hı hı.
2 minutes left, dear participants, for the speech of Nevzat Tarhan.
Yani bir radyo bağlantısı var Nevzat hocamızın. Bazen birkaç dakika uzayabiliyor. Birazdan aramızda olur.
By the way, I think we have problem with translation, hocam.
Yeah, I saw it.
So I think we cannot translate it, right?
Yeah.
Sorry. Dear newcomers, we are waiting for Professor Dr. Nevzat Tarhan. That's why we are not talking. Just let's wait 5 minutes. Then I hope he will attend as soon as possible. Herhangi bir bilgimiz var mı Nevzat Hoca hakkında? Programa bitmiş olabilir mi? Ne zaman katılır?
Bilgi alalım Şeyma Hanım.
Bekliyoruz. Teşekkürler.
Hocamızın TRT yayını bitti. Birkaç dakika içinde bağlanacak. Az önce görüştüm kendisiyle.
Peki, teşekkürler. Bekliyoruz.
Rica ederim. Evet, bağlanıyor hocamız.
Evet. İyi günler herkese.
Hoş geldiniz hocam.
Hoş bulduk.
Hoş geldiniz hocam.
Hoş
Nasılsınız?
geldiniz.
Teşekkürler. Siz nasılsınız?
Teşekkür ederiz. Çok
Başlangıç
sağ olun.
yaptınız
Geldiniz
herhalde.
hocam.
Evet. Başlangıç yaptınız. Değil mi?
Evet, biz bir hoş geldiniz dedik katılımcılarımıza. Sizin konuşmanızı bekliyorlar kendileri.
Öyle mi?
Evet.
Evet. Ben de bu özellikle 8. Biyomühendislik Günleri, bu biyomühendislik günlerinde biz ilk yaptığımız şeyi hatırlıyorum. 8 sene önce. İlk yaptığımız zaman da hatta Yıldız'daki biyomühendislik Yıldız'da çok şey. Yani devlet üniversitelerinin içerisinde en güzel Yıldız'da var. Biz üniversite olarak biyomühendisliği açtığımız zaman çok dikkatlerini çekmiş. Bize de geldiler. Onlarla bir ortak çalışmalar yapmıştık. Yıldız Teknik Üniversitesi'nin. Orada şöyle şimdi biyomühendisliği anlatırken o zaman baktım Yıldız'dakiler biyomühendisliği yani biyolojik boyutu sadece kimyaya indirgemişler gibiydi. Ben orada biraz ezber bozdum. Yani bir kimya çok önemli tabii. Kimya, yani biyomühendisliğinin özellikle ilaç üretiminde, çeşitli araştırmalarda önemli. Ama biyomühendisliğin bir de nörobilim ayağı var. Nörobilim ayağı var. Şimdi 2018'de Davos'ta bu yaptı. Yani neural network'ün önemi patlama yaptı bütün dünyada. Yani biyomühendisin biyoinformatik ayağı ve onunla ilgili nöroteknolojiyle birlikte biyomühendisliğin kullanımı biyomühendisliğe yeni bir alan açtı. Yani o dönemde o şeydi tabii. Yani çok biyomühendislikte olan kimyacılar ağırlıklı gidiyordu. Onun üzerine bunlarla ilgili biz bazı dersler de koymayı yapmıştık o zaman. Yani derslerde ya da araştırmalarda onu da göz önüne almayı önemsemiştik. Şu anda yani bilimdeki gelişmelerde yani biyomühendisliği sadece böyle bir ilaç sektörüne indirgemek değil. Yani biyomühendisliği aynı zamanda nörobilim ayağını da ön plana çıkarmak gerekiyor ki bizim farkımız burada daha çok ortaya çıksın Üsküdar Üniversitesi diye. Bununla ilgili çünkü zaten beyin dediğimiz beyninizin çalışma sistemi 3 türlü çalışıyor. Bak. Bir sinyal üretiyor beynimiz, elektriksel. İkincisi kimyasal ateşlemelerle çalışıyor. Üçüncüsü de bu mevcut şeyler içerisinde enerji akışı olurken bilgi akışı oluyor. Elektriksel akış, kimyasal akış ve bunlara eş zamanlı bilgi akışı. Ve bu beynimizde oluşuyor ve bu şu anda aslındaki yeni beyin çalışmalarında yani bence biyomühendislik kuantuma da biraz dokunmalı artık. Kuantuma. Neden? Çünkü yani yeni mühendislikte beynin nasıl karar veriyor diye beyni incelediğiniz zaman bir insan bir kararı verdiği zaman beyinde bir kişi karar verir ben bunu yapacağım diye. Fakat enteresan bir şey oluyor. Beyindeki elektrofizyolojik ölçümlerde kararı verdikten 300 milisaniye sonra P300 dalgası ortaya çıkıyor. 300 milisaniye. O zaman karar veren beyin değil. Beynimizin üzerinde bir kuantum beyin var. Ve buna bilinç diyoruz biz yeni bilinç çalışmalarında. Bilinç diyoruz. Yani beynimiz üzerinde evrensel enerjiyle bağlantılı bir kuantum beyin var. O da bize karar vermemizi şu anda yapıyor, etkiliyor. Etkiliyor. Karar verdikten sonra beyindeki elektriksel potansiyel ortaya çıkıyor. Ondan sonra elektriksel potansiyel devam ettiği zaman kimyasal dönüşüme uğruyor. Bu kimyasal dönüşümüne uğrarsanız o kalıcı bilgi haline geliyor. Yani insan bir bilgiyi ilk beyne kaydettiği zaman elektriksel olarak kaydediyor. Ama tekrar ederse onu yahut da çok güçlü bir duygusal yükle kabul ederse beyine kimyasal harflerle yazılıyor bu sefer. Elektriksel değil, kimyasal ve kalıcılaşıyor. Onun için beyin kimyasıyla ilgili çalışmalarda çok önemli. Şu anda işte bakın Elon Musk deney hayvanlarında yaptı. Yani beyin bilgisayar arayüzü çalışmaları yaptı ve maymuna oyun oynattı. Yani şimdi bunu aslında bir iş adamı mı yapacak biz? Yani üniversite olarak bizlerin yapması lazım. Ama sermaye onlarda, bizde fikir var. Yani ikisini bir araya getirmek Türkiye'de mümkün değil. Onun için kendi kendimize söylenip duruyoruz ancak. Ama orada Amerika'da öyle değil. O adam Elon Musk bir şeye karar verdiği zaman o işi en iyi bilenleri getirip toplarmış. Müthiş bir Ar-Ge kuruyor. Mesela bu Amazon.com'un sahibi şimdi dünyanın ilk birkaç zengininden birisi. Ben öğrenince şok oldum. Kazancının %80'ini Ar-Ge'ye yatırıyormuş adam. Ar-Ge %80 ha %3-5 falan değil. %80'i. Ve bu TED Talk'larda konuşulan bir konu. Yani onun için kaynak sağlam. Ama Türkiye'de maalesef o da son zamanlarda %3'e çıktı. Yani sen Ar-Ge'ye yatırım yapmıyorsan sen arkadaş ancak bilimin nesnesi olursun. Birileri bulur, sen oturup takdir edersin onu. Ama bilimin öznesi olacaksak biz arkadaş Ar-Ge'ye yapacağız yatırımı. Biz taşa toprağa yatırım yapan bir ülkeyiz maalesef. Bu tarım toplumlarında taşa toprağa yatırım yapılır. Bilgi toplumlarında tasarımlara yatırım yapılır. Kimse alınmasın, gerçek bu. Yani onun için burada biz bunu bir bilgiye yatırım yapılmadıkça biz ondan sonra yani kendi kendimize sen ben bizim oğlan kendi kendimize övünüp dururuz. Onun için bizim burada üzerinde durmamız gereken şey arkadaş biz yani hiç olmazsa öyle hani büyük bizim üniversite olarak yolumuza asfalt döşenmiyor. Yani biz her şeyi böyle adım adım böyle çilelerle yapmaya çalışıyoruz. Ar-Ge konusunda birçok girişimimiz oldu. Mesela Deep Learning projesi, yani derin öğrenme projesi, proje yazdık. 7-8 senedir hayır da demiyor, evet de demiyor. Ankara'da bekliyor. Daha o zaman 3 milyon dolarlık bir projeydi. Ya %40'ını biz çalıştıralım dedik. Buna rağmen olmuyor. Ama bakıyorsun işte bazı konularda yani ondan sonra da mesela hatta ben onu hep eleştirdim. Yani yani 10 sene önce Fatih projesi oldu. Tamam, güzel, yenilikçi bir proje. Herkese tablet dağıtacağına 1 milyon tablet dağıtacağına 1000 kişiye yazılımcı yetiştirseydi şu anda kendi dijital platformumuzu daha kolay yapacaktık. Yani bunlar bence hep vizyonsuzluk. Vizyon nedir? İnsanın olabileceğini tahayyül etmesi. Misyon nedir? Yapabileceğini tahayyül etmesi. Olabilecekleri önce belirleyeceksin ki neler olabilir? Hayal kuracaksın. Ondan sonra bir misyon, sahip olduğun şeylerle en iyisini yapmaya çalışacaksın. Onun için biz yani üniversite olarak böyle bir vizyonumuz, stratejimiz var. Yapabildiğimiz kadar yapıyoruz. Yapabileceklerimiz yani olabileceklerimizle yapabileceklerimiz arasındaki mesafe çok açık da olsa biyomühendisliği ben daha Türk bilim dünyasının çok tanıdığını düşünmüyorum. Önemini fark ettiğini düşünmüyorum. Ama bu Covid ciddi bir şekilde bize biyomühendisleri önemini çok ciddi şekilde öğretti. Söyleyeyim. Yani moleküler biyoloji ve genetikle biyomühendislikteki öğrencilerimiz mezun olur olmaz. Şimdi yani mesela bizim hastanedeki biyomühendislik mezunu yanılmıyorsam.
Evet, evet, evet.
kullanan
Daha hızlı işe girmeye başladılar hocam bizim
Tabii.
mezunlarımız. Mezun
Tabii.
olmadan işe girenler bile olmaya başladı yani.
Başladı çünkü şu anda sağlık yatırımları dünyada birinci yatırım oldu ve bu konuda yani biyomühendislik konusu da bu ikisinin sentezini yapılacak biyolojik. Zaten insan bir varlık değil ki. Yani evren dijital bir evrendeyiz. Evrensel veri tabanı var. Evrensel veri tabanının üzerine bu bilgi var önce. Ondan sonra matematik var. Ondan sonra geometri var, tasarım var. Ondan sonra enerji, fizik var. Ondan sonra kimya geliyor, madde. Ondan sonra da biyoloji, canlı geliyor. Bunlar hepsi birbirinin halkaları. Yani birbirinin içerisinde olan şeyler. Böyle durumlarda biz yani insan madde diye bir şey yok aslında. Madde bir enerji yoğunlaşması. Madde dediğimiz şey. Yani kimya, yani ben onu biz hastanede ilaç kan düzeyi bakıyoruz. Türkiye'de ilk defa biliyorsunuz. Yani şimdi bizim üniversitede laboratuvar onu hastaneye aldık, sonra buraya taşıdık. Terapötik drug monitoring, orada LC-MS cihazı var. O LC-MS cihazı 3 tane var. Biri toksikoloji, maddede ölçüyor. Diğeri işte ilaç kan düzeyi, psikiyatrik ilaçların 2 tanesi ölçüyor. Biri yedek gibi. Şimdi bunlar çalışırken ben hani merak ettim bu konuya. Yılda 2008'lerde falan araştırdım. Ya madde dediğimiz şeyin mesela ilacın kandaki düzeyine bakıyorsun. Her ilacın bir iyon yükü var. İyon yükü. Yani ilacın iyon yükünden dolayı o ilacın kütüphanesi o maddenin olup olmadığına karar veriyor. Mukayese yapıyor. Bu iyon yükü tabii orada sistem onu 2 defa doğuruyor MSMS denilmesi. Yani klasik o şeylerden değil. Likit kromatografi yani likit yöntemlerden değil. Bu şeylerle ilgili. Yani e, e, böyle double check yapıyor sistem. Double check yaptığı için de iyon yükünü kütüphanesine bakıyor. Bu iyon yükü filanca maddeye uyuyordur diyor ve oradan bu o maddenin olduğunu anlıyoruz biz. O madde olduğunu. Mesela diyelim et parçası geliyor. Bunun domuz eti mi olduğunu, dana eti mi olduğunu, koyun eti mi olduğunun özel bir şeyi var onun. İyon yükü var proteinin. Onu belirliyor, söylüyor. Bal, hakiki bal mı değil mi? Pat diye söylüyor. Enteresan. İlaç o mu değil mi? İyon yüküne göre söylüyor. Ve bunlar o kadar kesin ki yani DNA'nın doğruluğu nispetinde doğru sonuç veriyor. Yani o derece %99 üzerinde doğruluk sonuçları veriyor. Bütün bunlar kimin işi aslında? Yani biyomühendislik işte bak beyinle kimya ile teknolojinin ortak kullanılmasıyla ilgili alanlar. Yani bu alanları bilen kişiler şu anda geleceğin mühendisleri olacaklar. Yani burada kimisi kimyaya odaklı ilerleyebilir, kimi nörobilime odaklı ilerleyebilir. Kişiler bunun kararını kendileri verecekler. Yani böyle alt disiplinler olur devamlı bunun altında. Yani birlikte biyolojik bilimlerle mühendislik bilimlerinin birlikte yapması gerekiyor. Ve ortak multidisipliner çalışması gerekiyor. Zaten şu anda da bilimlerin böyle tekilliği değil, bilimlerin çoğulculuğu ve bütüncüllüğü ön planda, eklektik yaklaşım ön planda. Multidisipliner yaklaşımlar, bilimlerin böyle takım halinde çalışması yeni keşifleri ortaya çıkarıyor şu anda. Onun için biz biyomühendisliği ilk başta kurarken yani yani hastanelerde, ameliyatlarda vesaire doktordan çok gelişmiş ülkelerde mühendisler var. Yani tahmin ediyorlar, seçenekler sunuyorlar ve işleri daha kolay, daha az hatasız yapıyorlar. Yani onun için bunların biz geçirilmesi gerekiyor. Yani hayata geçirilmesi gerekiyor ama Covid gibi bir tokat yememiz lazımmış bunu anlamak için. Yani, insanlık olarak yani Covid tokatı zihinlerde ezberleri bozdu. Zihinlerde sadece işte mühendisliği, sadece maddesel böyle materyalist bir bakışla baktırmıyor. Artık bütün disiplinlerin birlikte olduğunu, bütün bilim dallarının takım halinde çalışması gerektiğini daha çok ortaya çıkardı. Yani mesela bakıyorsunuz Covid'le konuşanlar hep enfeksiyon hastalıkları uzmanları konuşuyor. Enfeksiyon hastalıkları uzmanları hangi konuda konuşur? Hastalığın yayılması, bozulması, ya hep tehdit ayağıyla konuşuyorlar Covid'in. Ya bir kriz varsa, bir tehlike varsa onun bir ayağı tehditse bir ayağı fırsattır. Bu bize ne kazandırdı? Bireye ne kazandırıyor? Topluma ne kazandırıyor? Gelecek bununla ilgili yani yıkıcı inovasyon diye bir kavram var şu anda bak. Yıkıcı inovasyon. Yani yıkıcı inovasyonu ilk ne zaman başlıyor insan hayatında biliyor musunuz? Doğumda başlıyor. Çocuk annenin karnının konforlu ortamında gayet güzel yaşarken birdenbire ağzını bile oynatması gerekmiyor yani. O kadar rahat, konforlu bir ortam ki. Fakat doğuyor. Doğduğu zaman birdenbire soğuk hava ciğerlerine giriyor. Birdenbire müthiş bir korku hissediyor. Korku hissediyor. Hemen o korkuyla ilgili anneye sarılıyor. Rahat kokusunu alıyor, rahatlıyor ve güvenli bir alan oluşturmaya başlıyor. Anneyle sıcak ilişki olduktan sonra çok yavaş yavaş hayata alışıyor. Ama o çocuğun ilk andaki şeyi böyle tepkisi müthiş bir dehşet ve korku halidir o çocuk. Boşuna ağlamıyor çocuk. Ama o doğum bir nevi farklı bir banttan, enerji bandından farklı bir enerji bandına geçmiş gibi oluyor. O doğum onun için yıkıcı, sarsıcı bir şey ama onun için bir aynı zamanda öğrenmedir. Ne oluyor? Hayatın gerçekleriyle ilk başta karşılaşmaya başlıyor. Öğrenmeye başlıyor. Bunlar yıkıcı inovasyondur. Yani şimdi şu anda Covid'de küresel olarak Great Reset deniyor hatta bunu. Amerika'daki felsefi tartışmalarda büyük reset, büyük sıfırlama. Yani her şeyi yeniden baştan yazacağız. Önem ve önceliklerimiz değişecek. Mesela diyor, o zaman Amerika'nın önem öncelikleri neydi? Hedonizmdi. Hollywood kültürüydü. Eğlence hayatıydı. Gece hayatıydı. Müthiş. Ona doğru haftanın 5 günü çalış, iki gün eğlene odaklıydı. Bunları şimdi yapamıyorlar. Yani böyle durumlarda yani bu yeni pozisyon almaları gerekiyor artık. Bu yani şu andaki yapılan tahminlere göre, matematik modellemelere göre 2024'te ancak normalleşme olacak. Ders olsa bile maskeli olacak, mesafeli olacak seneye. Yani onun için böyle yani öyle kolay normalleşme, eski hale gelme olmayacak. O halde yeni duruma uyum sağlamak için bu olayı bir yıkıcı inovasyon olarak görüp buradan yeni şeyler üreterek çıkmak, insanlık bunu yapabilirse kazananlardan olacak. Bunu yapamazsa kaybedenler olacak. Tarihin çöp sepetinde kalacak. Mesela Osmanlı endüstri devrimini neden kaçırdı? Endüstri 1.0 oldu, makine devrimi. Ondan sonra Endüstri 2.0 oldu işte buhar makineler diye. Endüstri 2.0 elektrik çıktı. Ona da önemli bir gane kaldı. Endüstri 3.0 çıktı. Elektronik devrim oldu. Daha çok 20. yüzyılda oldu. O zaten biz Cumhuriyet'te kaçırdık onu, Cumhuriyet döneminde. Ama o dönemlerde hatta medreselerde eğitim verilmek istendi. Medreselerde sadece din eğitimi veriliyordu okullarda. Bu matematik, fen bilimleri eğitimi verilmek isteniyor. Ahmet Cevdet Paşa var biliyorsunuz şu anda. Mecellenin yazarıdır adam ve hem doğuyu hem batıyı bilen süper paşalardan birisidir. O bakıyor ki ya biz sanayi devrimine karşıyız. Osmanlı ilk defa borç alıyor. 1854'te ilk defa borç almış. Çünkü artık öyle değişmiş. Halktan toplanan vergilerle devlet yürümüyor. Onun için onun için bakıyor ki şu anda Cağaloğlu Lisesi'nin olduğu yerde bir açıyor. Cağaloğlu Lisesi'ni yaptırıyor ve oraya Medrese-i Zülcenaheyn diye ismini tanif ediyor. Bunun son temsilcisi Erzurumlu İbrahim Hakkı'dır Marifetname'de. Mesela astronomiden filan bahseder orada. Astronomiden, matematikten bahseder. O Osmanlı bunu 1580 tarihinde bu olumsuz dönüşümü yaşıyor. 1580'de ne oluyor? 3. Murat bir şekilde Mısır'dan gelen o Yavuz'la birlikte gelen ulema, bunlar şey karşıtı, akıl karşıtı ulema. Akli ilimler, zahiri bilimler, batın ilimler, akli bilimler ve nakli bilimler gibi bir denge var. Bu akli bilimlere tamamen karşı bir akım bu. O akımın temsilcileri İstanbul'a geliyorlar, Osmanlı ulemasını etkiliyorlar. Ve o zaman Fatih zamanında kurulmuş Ulu Bey Rasathanesi var, tam Tophane'de. Yani Ulu Bey Rasathanesi'ni Takiyüddin Efendi yeniden yaptırıyor, çok eskidi diye. 5 sene içerisinde böyle bazı hastalıklar, böyle musibetler oluyor. Onun üzerine o ulema diyor ki, Allah bizi göğü izlememizi yapsa gözümüzü ona göre yapardı. Allah bizim uçmamızı istese bizi kanatlı yapardı diyerek bu padişahı etkiliyorlar. Padişah da Tophane'deki rasathaneyi bombalatıyor. Bunu teyit ettim ben, bombalatıyor. O zaman Osmanlı zaten zengin olmanın 1580 Kanuni'den sonra yani şey yok artık, yani dünyanın tek süper gücü durumunda. Böyle durumda da çok ihtiyacım da yok diye şeyleri yavaş yavaş terk ediyorlar. Sadece din eğitimine odaklanıyorlar. Ahmet Cevdet Paşa buna fark ediyor bunu. Yani astronomiyi, fiziği, felsefeyi, mantığı, Aristo'yu, bunları, matematiği ders olarak koyuyor. Duvarlarda haritaların olduğu bir medrese yapıyor. Adına da Medrese-i Zülcenaheyn, 2 cenahlı medrese diyor. Sonra o zamanki vakıf uleması medrese eğitimi onlardaymış. Hemen hemen Osmanlı'da bir dönem Osmanlı'nın %16'sı vakfın elindeymiş. Düşün. O derece büyük bir medresede de tekel oluşturmuşlar. Medrese uleması şiddetle karşı çıkıyor. Padişah da mecbur kalıyor, 1. Abdülmecit. Kapattırıyor orayı. Endüstri devrinin boşuna kaçırmamışız biz. Onun için bunlarla yüzleşmemiz lazım arkadaş. Osmanlı tamam atamız bizim ama hatasız değil ki. Ondan sonra bakıyor bunu Abdülhamit mesela çok akıllı birisi. Bunun farkına varıyor. Ya bakıyor bu medrese bu gidişle kendi kendine çöpe gidecek. Tarihin çöplüğünde kalacak diyor. Kendisi sultanileri kuruyor. Sultanileri kurduğu zaman işte askeri, tıbbi, şahaneyi kuruyor. 2. Mahmut da öyle mesela. Bunları kuruyor. Böyle bunları kurulduktan sonra orada insan yetiştiriyor. 2 tip insan yetişmeye başlıyor bir seferde. Biri Batı bilimlerine, yeni bilimleri yücelten ehli mektep, diğeri de sadece din bilimlerini yücelten ehli medrese. Bu ikisi birleşiyor. 2 tip insan yetişiyor. Zamanın ruhunu ehli mektep dediğimiz kısım yakaladığı için onlar ne oluyor? Abdülhamid'in kendi yetiştirdiği çocuklar kendisini yıkıyorlar. Daha bir sonraki kuşakta Osmanlı yıkıyor. Halbuki Osmanlı meşrutiyeti kabul etseydi meşruti monarşi olacaktı ve böyle durumlarda yavaş geçiş yapacaktı. Yani Cumhuriyet döneminde de tabii bunun üzerine tam tersi bir tepki oluyor artık. Yani ifrat tefrit arasında kalmışız. Sentezi yapamamışız sonuçta. Neyse konumuza direkt ilgili değil ama yani yenilikçilerle yani vizyon yıkıcı inovasyonlar bize bir şey öğretsin. Burada da bazı şeyleri yeniden düşünmemiz, yeniden yapılandırmamız, yeniden keşfetmemiz gerekiyor. Covid bence biyomühendisliğin, moleküler biyoloji, genetiğin önünü açtı ve açacak, öyle gözüküyor. Bunu bu gözle değerlendirelim. Yani arkadaşlarımız bu konuda daha böyle geleceklerdeki öğrenci arkadaşlarımızın da muhakkak böyle öğrenci projeleri yapsınlar. TÜBİTAK vesaire öğrenci projelerini destekliyor. Öğrenci projelerin yapısından hocaların danışmanlığında o projelerle emin olun çok şeyler böyle. E, Yani üniversite lisans döneminde proje yapmak var ya, o geleceğe farklı bakışı sağlıyor. Onun için yani şu anda da zaten üniversitelerdeki rekabette ciddi bir rekabet var şu anda, vakıf üniversiteleri. Yani devlet üniversitelerine değil, öğrenciler vakıf üniversitesine gidiyor diye maalesef vakıf üniversitesi tırpanlanıyor. Mesela ikinci öğrenimi bizim 40 kişiyle sınırladılar öğrencileri falan gibi böyle. Yani böyle Anadolu'daki üniversiteler boş kalıyor diye. Ya bunun sebebini düzeltmiyorlar. Sadece öğrencileri Anadolu Üniversitesi'ne sevk etmek için. Ben şey yapıyorum, ben biraz doğruya doğru, eğriye eğriye diyen bir tipim. Yani kimse üzerine almasın. Ben hiçbir kişi şey yapmıyorum ama gerçekleri de konuşmazsak ben bilimsel kimliğe uymamış olurum. Yani bilimsel duran objektif durur. Objektivizmin bilimin en büyük özelliğidir. Her şeye karşı objektif olacağız. Ama Mevlana'nın bir sözü var. Dost acı söyler ama acıyı tatlı bir şekilde söyler. Böyle söylemeye çalışıyorum. Bu kadar iğneleme yeter herhalde. Yani, ama ben biyomühendis ve moleküler biyoloji genetikteki arkadaşlarım, bu sentezi yapan arkadaşlarımın gelecekte daha güzel şeyleri yapabileceklerini düşünüyorum ve başarılar diliyorum.
Çok teşekkür ediyoruz hocam.
Estağfurullah.
Teşekkür ediyoruz hocamıza konuşmaları için. Thanks for speech of the directors. I think we can continue with Dr. Hasan Demirci. Before his speech, I want to give a brief information about Dr. Hasan Demirci. Professor Demirci completed his doctorate at Brown University, and he currently continues his studies at Koç University, laboratory researchers, and he has a lab at Stanford University again. Today, he will talk about his study about structural biology, and his work area is X-ray crystallography studies of ribosome complexes. I'm giving the floor to Dr. Hasan Demirci. Please, Doctor, we are listening to you. Thank you.
Thank you very much for the kind invitation, Şeyma Hocam. I just wanted to say that I'm blown away by the quality of the students that Üsküdar University graduated. I have two students from your university, two recent graduates, Ebru Destan and Gözde Karakadoğlu. So they're doing PhD with our group. I am basically the leader of Koç University Structural Biology Knowledge Center and also the Innovation Center, Koç Üniversitesi Yapısal Biyoloji ve İnovasyon Merkezi. And I just started at Koç 18 months ago. So we have an 18-month-old baby. And I'll talk about what we've done in Turkey for the past 18 months. I could have listened Nevzat Hoca all day. So he said a lot of good stuff. And I fully agree. I think a lot of people see the COVID-19 as a disaster, but every disaster comes with the opportunities. And here we'll talk about what we've done during this hardship, how we built a lab twice from scratch at Koç, and also how we executed some of the most difficult to, technically challenging experiments toe-to-toe with the international groups. So there's a great competition here. And I think, what's really important here is that the question, can we do good science in Turkey? I'm going to change the question. Can we do amazing science in Turkey? So I think if we can put our resources together.
I think we have connection problem, right? Or only me?
No, we have.
We have a connection problem.
Let me text the doctor. Doctor, we have connection problem, I think.
Can you hear me? Like, where did I
Yes.
left?
Yes, now yes.
You said about in Turkey, sign. Can we
Yeah.
do signs in Turkey like that?
Yeah, can we? Yeah, the most important $1 million question is that can we do good science in Turkey? And I'm going to change the question. Can we do amazing science in Turkey? The answer is yes. We can do amazing science in Turkey. And in order to do that, there's only one condition. We must want to do amazing science. We can do the most amazing science if we want to do it. And what I'm saying here is that we don't want to. It's not like we cannot. If we can come together, we can do it. What is it that we cannot share? What is it that, holding us back? I have my saying, 1 Turk is better than 10 Japanese in terms of innovation, in terms of, hard work. But there is a comma. That's not the full stop of the sentence. But 10 Japanese people can do better work than 1 million Turks. They can come together. They can work together. They can do amazing things together. And we cannot. The 2 Turks cannot come together. So here at Koç University, we have a laboratory. It does not have anywhere have my name on it. It's Koç University Yapısal Biyoloji ve Innovation Center. And here is our laboratory. There's nowhere my name. And I love to work institutionally. It's all about coming together, all right? It's all about sharing. And you cannot lose your information by sharing it. You can only increase the knowledge. And here, what we're trying to do, foster a laboratory culture where our students can come together. Doesn't really matter what kind of political view they have, what kind of background they have. They're all welcome and they're working together and they're the team, and their success is their success. Their failure is my failure. I take full responsibility. And I think, just talking about science is amazing, but I just wanted to say that in order to do amazing science, you should have an amazing team. And you have to be surrounded by amazing people. And we have to cultivate that culture. And if you let me, I would love to share my screen and talk about what we've done in the past.
That will make you the host.
Yeah.
You can share. Okay.
All right. So the thing is that, can I have a magic wand and change the world? Can we do disruptive science? Can we create disruptive technologies and not compete? If we compete, we will lose. Period. We cannot compete with any of the Western machines. We have to reinvent the way we do science because we do not have the technologies. So what we're going to do, we're going to look at things from a different perspective. So we're going to create and reinvent the science the way we do it. For example, Nevzat Hocam talked about the drugs, right? And here is a hard fact. In Turkey, we do not have a single drug where we own the intellectual property. 0, none. It's a fact. We can only do generic drugs. We cannot do anything bio-better. All of our drug companies are trying to do biosimilars, not bio-betters. They're trying to imitate, they're trying to do something that has been done 100 years ago. We cannot even produce an insulin, a recombinant protein that has been done decades ago. So what are we going to do? How are we going to change this? And at least I can talk on my behalf. The way we're going to do science in Turkey will be different. I kind of closed a laboratory at world's one of the the best university, all right? At Stanford University, I closed a shut down a lab and brought it into Koç University. Now we have the most, I would say, competitive, internationally competitive, one of the top of the line, the state of the art, beyond the state of the art laboratory where we can do cutting edge science at Koç University, toe to toe. So today I'm going to talk about the bright future of the structural biology in Turkey. and I'll talk about ambient temperature, serial femtosecond X-ray crystallography studies of SARS-CoV-2 main protease. All right. I said these are the drug compounds, but in order to image biology at atomic resolution, we have to freeze these molecules and biology to minus 180 degrees Celsius. That's why none of the structural biology is able to kind of like produce any viable drug candidates. Now, with my group at Koç University, we can do these imagings at ambient temperature, at near physiological temperature, where biology is alive, where it's moving, where things are rotating. So we can image biology as it happens in front of our eyes. Instead of taking its pictures, we can be the Pixar studios of biology, and it's going to be the world's most amazing studio in the world. All right. So we have a beautiful campus, and we have a beautiful team, and we're going to do beautiful science. And we also have one of the most amazing infrastructures of Turkey. And I'm proud of being a member of Koç University. They never promised and they never under-delivered anything. I'm so grateful for everything they provided to us. This is no commercial. And here we can do amazing science. We can do a science better than I was able to do at Stanford University. And we have all the necessities that we needed. So this is our biosafety level 3 laboratory, one of the best in Turkey. And now I had the access to these facilities. And today I'll talk about the SARS-CoV-2. I know you guys all know what this thing is, the life cycle of this virus, why do we target the main protease of this drug, and how we're going to do it. And now you know that there's more than 150 million people sick, more than 3 million people dead. And when we started this work from Bahçeşehir University with Serdar Durdağ hocam, the idea was to do a quick drug repurposing. He screened all the available drugs that are targeting the main protease of the SARS-CoV-2, and we were going to do the structural biology and visualize these drugs bound to these targets proteins from the SARS-CoV-2. Things turn south, and we see this non-conventional drug development and vaccine development. When we started, there was no available vaccine. It was like April 2020. There was no drug. There was nothing available. There was a chaos. And we wanted to do something very quick. All right. And you guys all know the symptoms of this disease. You know how cute is this small virus. It's absolutely gorgeous. There's only like 20,000 nucleotides. And there are only a few proteins on its surface: envelope, spike protein, membrane protein, nucleocapsid protein. And our goal here was to target not one of these surface proteins, but our idea was to find a compound that can go and target a drug, a target in the virus that does not exist in the human body. And we chose to target main protease because this virus first synthesized a polypeptide, 2 of them in 2 open reading frame. And then once it has these like polypeptides is synthesized, there is either papain-like protein or the main protease that comes and cleaves these peptides into smaller proteins so that the virus can reassemble. I'm going to skip these. And in the life cycle, after the entry and the synthesis, the virus must proteolyze these long peptides into smaller proteins so that can reassemble. The good news is that there is no such protease in our body can go and get targeted by the drugs that were targeting the active site of this protease. So the goal was to find a drug and then look at the binding and then test it structurally to see how this drug binds and how we can just kind of like target these. And Serdar Hoca's one of the drugs, Montelukast, right now in clinical trials. And this was a study that we started like a year ago, almost a year ago. So as you can see, if we can come together, things can go very fast, faster than all the other laboratory in the world. I think this is going to be one of the first drugs that are kind of like targeting these SARS-CoV-2 virus. So there's cytokine storms. I'm not going to talk about it. And the treatment, when we started, there was no FDA-approved treatment or the vaccines. Now we have it. So what we learned from this experience is that there is no infrastructure in Turkey that we can do the structural biology research. So again, my question, can we do amazing structural biology in Turkey? The answer is again, yes, because we wanted to do structural biology in Turkey, not in Europe. In order to do that, we had to write proposals to the US facilities, and we secured beam times, and we executed some experiments. We use X-rays. X-rays has the low wavelengths that allows us to see the atoms, but when we have the short wavelengths, that means it's increased energy. These are ionizing radiation. When you wanted to see things with the X-rays, that basically vaporizes in front of your eyes. So we have to cool it down so that we can image biology at minus 180 degrees Celsius. So in order to get the images, we need the crystals of the biological specimens, either proteins, DNA, RNA, and we heat them with the collimated X-ray beam, collect a two-dimensional diffraction data from the crystals. So in order to do that, we grow the bacteria, we break open them with the sonication or emulsifying. Then we break them open and purify them with chromatography, and we set up them for crystallography experiments, and we obtain beautiful crystals, and there are more of these. And once we have the crystals, a human eye has a lens that can focus the visible light to a retina, so we can see the image directly in our visualizing center, occipital center. But with the X-rays, there is no lens. We can focus the X-rays. So we need these two-dimensional detectors to collect the data, but we cannot focus for direct imaging. So what we do is we use algorithms, and we use not artificial intelligence or machine learning, we use natural intelligence, all right? So what's in this box over here is basically the essence of X-ray crystallography. I'm just gonna show a brief picture of what's inside this Pandora's box, all right? So this is X-ray crystallography. After we crunch these numbers, we basically reconstitute a three-dimensional electron clouds around the atoms, all right? And then we start building our models. These are not calculations. These are experimental maps showing the positions of each atoms. And X-ray crystallography is the most powerful, the most robust imaging method. And here you can see even a water molecule, and there you have the small hydrogen atom, one of the smallest atom in the world, all right? Then after we built the main chain, we built all the side chains, and then we have an atomic model where we can then model these drugs and how they interact with the target so that we can design a bio-better or the better next generation, second, third generation drugs that can target these proteins and also the other targets. How are we gonna do this imaging differently? Here is at Stanford, first hard X-ray linear accelerator where we do free electron laser. And I used to have a laboratory over here, which I shut down, put it into a container, and kind of brought it, teleported into a Koç University. I'll explain how we done that. And in this machine, we can do imaging at ambient temperature without freezing samples in liquid suspension as it happens in physiological conditions, all right? So we reinvented the crystallography with my kind of like the young colleagues. And in our group, we developed these $10 piece, it's a plastic piece, turned into this $1 billion microscope, 10,000 times more efficient. And we published this in one of the most prestigious journals. So I just wanted to show how this machine works. Can you hear the sound, by the way?
The SLAC National Accelerator Laboratory
Yes, we
is
can.
located in the heart of California's beautiful San Francisco Bay Area. Operated by Stanford
So here's
University
a
for the
linear
U.S.
accelerator.
Department of Energy,
We
SLAC
have a
has been home to
synchrotron.
the world's longest
We have 8
particle
electron
accelerator
microscopes
for nearly 50
sitting there.
years. In 2009, SLAC ushered in a new era in its long history of physics research with a new kind of laser called the Linac Coherent Light Source, or LCLS. The LCLS is the first laser in the world to produce hard X-rays, which can be used to see down to the level of atoms and molecules. Adding almost
So
half
this
a mile
machine
onto the
uses
original 2-mile-long
16.2
accelerator facility,
gigawatt of
the
peak power
LCLS
that
uses the final
corresponds to
one-third of the
8
accelerator to produce
basically
powerful
Shimshek
pulses
lightning
of
per
X-ray laser
second
light.
to run this machine. $1 million
Scientists at SLAC
electricity
and from
built per
around the
day.
world use these powerful beams to explore how atoms and molecules move and behave on some of the shortest timescales imaginable.
So there's a drive laser here. I'm gonna fast forward these.
The
This
LCLS
laser
starts with
hits
the drive
into a
laser,
copper anode
which generates a
and
precise
release
pulse
the
of ultraviolet
electrons
light,
from here,
seen
from the
here in red.
copper plate. And
The
these
drive laser
electrons
pulse travels
will
down
be
to the
accelerated
injector
from the
gun, where it
furthest
strikes
speed, the speed of electrons, to speed of light in this linear tunnel at 16.2 gigawatt of
the
electric
electron
field.
bunch. Then
That's
encounters
where
the
the
second bunch
electric bill
compressor.
is coming
The second
from.
bunch compressor
And
is
these
longer
electrons,
than the first,
when they are
because the
traveling at speed
electrons
of
in the
light, they
pulse
lose
now
their
have even
charge
greater
and they
energy.
become chargeless. So we can collimate them, we can bring them together, and we can generate X-rays 1 billion times brighter than any of the other light sources. So this will be
The electron
basically the
pulse
brightest
continues
light
to the
source
end of the accelerator
ever.
at nearly
And
the
after
speed of
this
light,
acceleration,
finishing
the
the boost
electron
phase
goes
of its
into
ride.
undulator tunnel, and they
Magnets,
wiggle
which
here,
comprise
and they
thousands of
emit
alternating
X-rays.
north-south
Again, I'm gonna
magnetic
go fast. Here we can create
in tune
femtosecond
with each other,
pulses.
or
And these
coherent,
pulses, light
with an
can go to
enormous
moon and
boost in
back in
X-ray
1
power.
second. In femtosecond, the light can only travel the thickness of a hair. So the time will
Once the
stand
X-ray
still,
laser light is generated,
and basically
the
we
electrons
can
must be
collect
safely
data
discarded before the X-rays
faster
can be
than
used for
speed of light.
experiments.
How are we going to do
The
that?
beam
In
dump
1
uses
second,
a powerful
this machine
electromagnet
can
to
generate
divert the
120
electrons down
pulses.
to a special chamber
And now we're
that
separating the
absorbs the
electrons
electrons
from
and
the
dissipates
X-rays.
their energy.
So we're going to image 120
The X-ray pulse, unaffected
basically
by the pull of
image
the magnet,
per second from the
continues
biological
on in a straight
specimens
line.
without causing any radiation damage. So there are
This process
6
happens
end
up to
stations
120
in this,
times per
facility
second.
to do the experiments that we wanted to do.
The X-ray laser pulse is now ready for scientists to use in one of the 6 LCLS experimental stations. The experimental instruments each comprise a suite of vacuum chambers,
So these are the
detectors,
chambers
and
where we
sample
execute these
environments.
experiments. I brought a
Each
vacuum
instrument
chamber
performs different
and
kinds of
installed it
experiments,
at Koç University. Right
investigating
now
the
we have one
kinds,
here, so we can do
arrangements,
the
and motions of the building
preliminary
blocks of
studies in
matter.
our laboratory. And what happens here?
For example,
we introduce
the
the
LCLS
samples with an
pulse can
injector,
be used to make
and we
images
intersect
of
with
single
them
molecules,
X-rays.
even though the beam is
These
powerful
are
enough to
X-rays
instantly
brighter than the
disintegrate
sun's
such a tiny
plasma
sample.
temperatures.
Each
So
pulse
we
is so
immediately
fast
vaporize
that
the
an image is
samples,
captured in the
but
sliver
by
of
the
time
time the
before the
X-rays go
molecules
in
can fly
and get
apart.
out of the sample where everything moves at electron
Images
speeds,
captured in this
can
way
collect
can be
data
strung
without
together frame by
radiation
frame
damage and
to
construct
create
these
molecular
three-dimensional
movies
models
of individual
without
biological
any
molecules in action.
interruption. They're at physiological temperatures,
The
and they can
LCLS, a
also
revolutionary new
be
tool,
visualized without any
changing
damage.
how
So this
scientists
is
study the
basically the best imaging in the world, all right? So the name of the method called diffraction before destruction. But when I started, it was destruction before injection. So we have to reinvent the way we introduce samples here. I'm not going to talk about the details, but it's basically a syringe pump flowing the crystals into the interaction region so we can collect data. So what have we done at Koç University in the past 18 months? After we built our laboratory, within the first few months, we basically opened some boxes and start doing experiments. And these young people basically executed the first experiment of its kind. All right, LCLS was shut down for a year for a makeup. It's coming with more pulses. But the director was saying the LCLS is back better than ever with the help of our on-site team and Koç University laboratory. All right, and this picture was taken in 2009, and we kicked off and started a new era at this machine. And we executed the first SARS-CoV-2 experiments in visualizing these drugs that Serdar Hoca was calculating. And we experimentally observed the atomic level resolution of binding interactions. And this is the director. And director was saying that the first experiment of LCLS2, a new era, as I said, this is reinventing the biology. We started a new era of structural biology. And with all of these young people, you're going to see that all the names are Turkish, pretty much. Esra, Büşra, Cengizhan, Aleyna. And these are the ones who basically solved the world's 8th wonder. This is a experimental electron clouds obtained from the SARS-CoV-2 main protease. This was a collaboration with, again, Bahçeşehir University, Serdar Hoca. And here I said, the future is very bright for Turkish young structural biologists. It's so bright, it's 1 billion times brighter than elsewhere, all right? And we can do this better than anybody else. What if I tell you that we don't even have X-ray diffractometer in Turkey? Where is the leadership? Where is the vision that Nevzat Hoca was talking about? Nothing. So we can do these faster than anybody else, but we have to collect the data at another facility. Sorry. So then we published our first paper, and this was like all these young people and also an international team, first and last authors, all from Turkey. Here we just not solved one structure, but we determined 2 crystal structures of the main protease. And we obtained electron clouds at ambient physiological temperatures, so we can do drug repurposing faster than any other group in the world. And again, we can even visualize a water molecule and oxygen in the active site of this main protease if we need to design next generation inhibitors for these SARS-CoV-2. Maybe not COVID-19, but COVID-21, if it happens, will be the first group coming with the drugs and vaccines, if possible. So what else we're going to do? We have a very versatile technology. We're not just collecting data at X-Files. We can do this at synchrotrons. We can do this at home sources, as simple $1 million machines that we can purchase in Turkey. And also, we're also switching and doing more cryo-EM single particle imaging. I'm not going to talk about details, but we are doing a multi-scale imaging. So now, this machine that I just showed you was working at 120 pulse per second. Now it's taking a new turn. There's going to be a new upgrade to this machine, and it's going to be faster and better than ever. So now I want to show you the future. So far, I show you the past, and now we will see what's going to happen in the future at Koç University. So our laboratory here, now at Koç University, I'll show the pictures in a while. So this machine was one of the world's engineering marvels, and our group get the best data from these machines, and now we're switching another time, and this is the undulator hole. We can see electrons moving from one atom to another because we have femtosecond pulses. 1 billionth of a millionth of a second we can make observations and imagings, and we can see the lights splitting the water in photosynthesis. We can do material science experiments, but it's all about making leaps, not incremental science. And this machine is switching from that copper RF to a superconducting technology, and that means there's going to be brighter light source, and there's going to be more pulses from 120. We go to 1 million pulse per second. The time scales, the resolution, the more data is coming for more discoveries, and we're going to do amazing science, and we're going to do it in Turkey, and we're going to do it at Koç University. And here is many of these people that we work with, all of these young people. Ebru is also from Üsküdar, and Serdar Hoca and his team. I'm supported by TÜBİTAK, and now I just want to show you what happened in September 2019. This is what I started with, and these machines all came from my Stanford laboratory in US. And then we opened the boxes, and then we start working immediately the next day. And then we started recruiting some students. And then we put some jackets, lab jackets, and some undergrads started working in the laboratory. This is an iGEM team working on synthetic biology experiments. Everybody's smiling. And we have the world's fastest cameras. Everybody is doing amazing science, and group get expanded and more expanded. And we have now altyapı. We also have these young people coming to our laboratory before the COVID. And this guy asked so many questions when they're visited. I almost offer him a PhD position. All right, then Koç University said, Hasan, we'll build you an amazing lab. And here's the picture. I said, I love it. Let's do it. And then we break apart the whole lab again, and we started cleaning. And then we start building a new laboratory with all the new equipment that we have. And now we have 34 seats for 34 young and talented scientists that want to do amazing science. And we also have a collaboration with Aselsan. We have world's one of the most amazing mirrors with the fastest camera available, where we did a lot of imaging. This is a manuscript also under review at a journal. And I would like to thank you for your attention, and I'll be very happy to answer if you have any questions. Thank you.
Thank you, Doctor, for this speech. It was really inspirational. for us. If you have any question, you can write on the chat part. Mehmet Akyıldız will ask to doctor. Yes, we are waiting. If you have any
Yes,
questions,
we are waiting
write.
for your questions.
They can email me. It doesn't really matter. I can even give my phone number so they can call me. We can talk about anything. What is your future plans,
If
hocam?
there's no questions, I can comment on something if you would like. Do I have permission?
Buyurun hocam.
Yeah, Hasan hocam, I am the next speaker here. But
Nice to meeting you, Tanıl hocam.
with this opportunity, I was very happy to listen to you, to your talk. And of course, we are not aware of these technologies very quickly, you know, what's happening in Turkey. I'm very happy to see that all is now in Turkey together with you. Of course, your experience is more important probably than these instruments only that you are building. I'm very happy to see another person like me, because in our university also there's no personal lab, personal equipment or anything. We started about 12 years ago in Acıbadem University. And I would like to invite you also to our university sometimes to see what is the, you know, the equipment, all the infrastructure here. But we started with the same, you know, understanding and policy here to share everything. And we are sharing everything. We have also, I believe, one of the best, you know, infrastructure for health sciences, for research around the world, I believe. So we are this not only with our students and staff, but everybody in Turkey. Lots of students are coming to our lab also as summer students from different cities and universities in Turkey, and we try to open as much as possible whatever we have here.
So, Tanıl hocam, we have like 18 months old laboratory. I have like almost 12 graduate students, 3 postdocs, and 5 MA students. So it's a group of 20. So usually they recommend like you make 1 kid every year, right? So it's just expanding 1 graduate student every year. But our group expanded so fast. And again, we promise that anybody in Turkey who wants to do structural biology, we don't even request any consumables. We give the consumables and everything, and we do everything. So we just want people to do structural biology. We have everything. We have enough resources that I brought from US. I can establish almost 10 structural biology labs in Turkey. We brought a lot of cheese. My students, when they establish their labs, I'll build their labs as well. So I really want this thing to be, you know, popular in Turkey so that, you know, we can design new drugs, we can design new pharmaceuticals, all right? And there was one question, what is the future plans, hocam? No plans, all right? The future is very bright and we're gonna be very disruptive. If I can tell you what the future is, then that's not a big vision. We're not trying to go to moon. If we try to do that, we're gonna fall back to the earth, go to stratosphere and come back. We're trying to go to Mars here. And we're gonna, worst case, we'll be end up at moon. And again, in Turkey, people are trying to do biosimilar. In our laboratory, we're only trying to do biobetters. And we will, worst case, make some biobetters.
Okay. So
Thank you,
hope
Dr.
to see you someday.
Thank you very much.
Together in our university.
Likewise.
Thank you very much for the
Thank
very
you,
nice talk.
hocam, for this exciting talk. It's very enthusiastic, and we hope to see many more researchers coming from your group. And we want more groups like you, actually, and Tanıl hocam's group. So hopefully we'll not have biosimilars anymore and biobetters. Something new, please. Thank you so much.
Thank you.
if there is no any question, we can skip the Tanıl Hoca, I think.
Yes, yes.
Thanks you again for the speech of the Hasan Hoca. Okay, we continue with the presentation of Professor Dr. Tanıl Kacagöz. Let me give a brief information about Professor. He completed his master degree at Hacettepe University. If I'm wrong, doctor, please correct me. Master degree at Hacettepe University Medical School in the field of microbiology. Completed his doctorate at Hacettepe University in the field of biochemistry. Okay? The professor has many awards and patents he has won. Now I'm giving the floor to Professor Kocagöz. Please, doctor, we are listening to you with your presentation.
Okay. I would like to share my presentation, if possible, Şeyma. It gives, okay. Oh. Okay. Okay, I think you can see the presentation, no problem. Okay.
Yes, we can.
Okay. I mean, when they called me, they asked me to talk about whatever you would like to talk about. Of course, everybody's interested in COVID-19 right now because of the pandemic, as you know. So I chose the laboratory diagnosis of COVID-19 to present it to you. So I will try to summarize it very quickly because I had only 20 minutes to talk about. But I will try to also tell you about what we are doing as research to support these kind of diagnostic techniques. So, as you know, viruses are very simple organisms. In fact, there's a nucleic acid, and then in the middle, and then there's a capsid, and we have sometimes a membrane that covers the viruses, and that's all it is. So when we do the diagnosis, we have to detect one of these structures, as you know. So, of course, the most common technique that we use right now for the diagnosis is you hear every day that we have done in Turkey, for example, about 300,000 tests with a PCR, polymerase chain reaction. So I will talk about this a little bit, and then newly there are some antigen tests and antibody tests that are introduced so that we can do maybe more rapid diagnosis of COVID-19. So in order to understand the sensitivity of these tests, of course, we should know when we have different kind of molecules belonging to COVID-19 at what time of the disease. So you see here in this picture, we have a viral RNA, virus itself, present in the respiratory secretions before about 1 week before the symptoms start in this disease. So if we do PCR at this time, we can detect the RNA of the virus. And then the antibodies start later, as you see, but we can also detect, as you see, some antigens, the proteins that are present on the virus that may be found in the clinical samples. If you want to isolate the virus by cell culture, you can do this within, 2 weeks after the onset of the symptoms. Before the symptoms, it is possible because there are replicating viruses there. But as you see here, this goes very quickly down because you cannot find the virus on the upper respiratory tract after 1 week after the symptoms are started. Now the virus goes to lower respiratory systems and to other parts of our body. So it is not, it's becoming harder and harder to find the virus in the upper respiratory system where we can get our samples easily. So I will not go, of course, into the detail of PCR. Probably everybody knows polymerase chain reaction, how we amplify the nucleic acids. But since this is a RNA virus, the genetic code is RNA, not DNA. So we have to start from RNA. And as you know, we cannot amplify RNA by PCR. So what we do, we obtain a DNA copy, complementary DNA copy, what we call cDNA, of starting with the virus RNA here. So after we obtain this DNA, now we can amplify the DNA by polymerase chain reaction, as you know, by denaturation, binding the specific primers, annealing the specific primers at a lower temperature, and then doing DNA synthesis at polymerization step. So, of course, in classical PCR, after we amplify, we have to run electrophoresis, the products, to see the amplified product. However, in real-time PCR, we can observe the DNA amplification at real time. I mean, it should be called, as I believe, observable PCR, not real-time PCR. In fact, this is a wrong name that was put originally, because everything is real-time in life, whatever you do. So this is, they mean, in fact, observable PCR here. So how do we observe? We use either some fluorescent labeled probes or some fluorescent dyes that bind to DNA and give fluorescence, give light during amplification. So, I mean, the earlier you see the amplification during these cycles, it means that you have a higher content of virus in your sample. Of course, I would like to remember here Kerry Mullis, who invented PCR, as you know. Unfortunately, he passed away in 2019. So it's been almost 2 years now. And in fact, nobody noticed much that, Kerry Mullis died. But, I cannot think how we could manage COVID-19 diagnosis if there were no PCR today. This is a book written by him, Kerry Mullis. You can find this in the internet, this book. But I would advise you to, read this book. It gives lots of interesting ideas how the scientific world works in United States and elsewhere in the world. I think everything is interesting in this book. So, of course, as you know, when we use PCR for the diagnosis, it takes some time because you have to take the sample and then you have to extract the RNA and then you have to do the PCR amplification, and you need a molecular biology laboratory set up to do PCR and some trained personnel, to do all these mixtures and do the tests and everything. So, but the isolation of the patient as early as possible is very important to stop the chain of transmission of COVID-19 in the population, as you know. So therefore, if we can develop more rapid tests, and even if it's possible to for the people to test themselves at home without going to the hospital, it will be much more important to stop the transmission of the disease. So we can use nowadays, I mean, more and more rapid antigen tests are coming. So these are immunochromatographic assays, what we call. In the strip, in a cassette like this, there's a paper strip where you apply your clinical sample. And then there are, gold labeled antibodies here, which are specific for the COVID-19 antigens, COVID-19 proteins. So if there are COVID-19 proteins, these bind together and start to run together, which are captured with another antibody here against this COVID-19 specific antibody. So they create a red line like this. You see here, the test line here. This is a positive sample. And of course, there is a control line here. If not even nothing binds to this, they will bind to the control line here so that you can understand the test is completed without a problem. So this test takes usually 5 to 15 minutes, to complete. And it's very easy to apply. However, unfortunately, the sensitivity of this test, since there are no amplification of the antigens here, is not as good as PCR. However, this is coming more and more closer to PCR sensitivity. The same way, of course, you can detect antibodies in the blood of a person with this kind of test. So if you put here, instead of, antigens, you can put labeled antigen, I mean, SARS-CoV-2 antigens, proteins here. So if there are antibodies that will bind this, now they will be captured again in this line. And you can use here on this membrane anti-immunoglobulin G antibodies or anti-immunoglobulin M antibodies. So you can also differentiate, you can also identify the presence of either IgG or IgM antibodies. Because first in our body, whenever there's an infection, at early stage, IgM antibodies are produced, then IgG antibodies will be started to produce. So IgM antibodies in COVID-19 do not last more than 2 months. So if you find IgG antibodies positive, but IgM negative, you will understand that the patient had the disease before 2 months. If these are both positive, this may be a 1-month-old infection, or if only IgM positive, then you will understand this is a new infection, which started about 1 week, 2 weeks ago. Of course, this doesn't give you, the chance to isolate the people because this gives results late. So what we need, the antigen test or PCR for rapid identification. You can, of course, use ELISA test, which is more sensitive than this rapid antigen test to determine the antibodies or antigens present, in the blood of a person. Since there's amplification of the signal here by enzyme-linked immunosorbent assay, you add a substrate of this enzyme that you fix here. So you can have better sensitivity with this kind of test. These are good tests in order to understand, what is the immunity in the population. So we vaccinate people or people have natural infection. But if you want to do a research, especially how many people are now immune against the disease, So it's a good way to take a, sample of blood from, selected volunteers and then look for the antibody if it is present against COVID-19. Since the antigen tests have no good sensitivity, we wanted to see if we can improve the sensitivity of rapid antigen tests by concentrating the biological fluids. So this is something that we have developed. In fact, we started this for the diagnosis of tuberculosis because tuberculosis is a disease that causes infection in the lungs, and it is very hard to take a sputum sample for the diagnosis. But the bacteria, Mycobacterium tuberculosis, that cause the disease, when it grows, we can see the antigens of this bacterium in the urine of the patients. And there are these immunochromatographic tests that can detect TB antigen, tuberculosis antigen in the urine. However, the sensitivity of these tests are very low. So we wanted to concentrate this by this polymer. So when you add this polymer into a urine sample, this will absorb the most of the water and small molecules, and high molecular weight molecules like proteins will be concentrated. So you here see the concentration of bovine serum albumin that we added into a urine sample. When we concentrate and analyze by electrophoresis, this is our dilute sample. When we concentrate once or twice, you see how the proteins can be concentrated. So we have seen that we can create a low molecular cutoff for the concentration. So anything more than 0.5 kilodalton can be concentrated with this polymer. You see here a molecular weight marker of proteins. The lowest band corresponds to 9 kilodalton here. And when we concentrate by these polymers, even 9 kilodalton can be concentrated pretty well. You can also concentrate DNA or RNA, whatever the nucleic acids are large molecules. When we do concentration with this polymer, you see here, again, concentrated 1 time or 2 times. This is the diluted sample. This is the original sample. And these are even 100 base pair, long DNA can be concentrated efficiently with this method. So we applied this for the diagnosis of tuberculosis. We got patients, TB patients, urine samples. You see here, unconcentrated and concentrated sample. This is a patient which doesn't have TB in it. So concentration did not affect, did not create any false positive result for us. But if you can see here, there is almost no band here. However, it becomes positive when concentrated. And in this patient, this faint band becomes clearly visible after the concentration of the urine. So when the test now became very easy to apply, so you put 10 milliliters of urine in your test tube and just dip this immunochromatographic strip in the tube. The antigens will migrate. If there is TB, you will see this band coming, from the patient. So it is a very quick, rapid test that you can do everywhere for the diagnosis. So when we started, when we were doing this research last year, COVID-19 came on. And as you know, for samples, now we are taking the samples currently by a nasopharyngeal swab. So theoretically, it looks very easy to do this experiment, but I don't know if anybody gave this kind of sample. You know how hard it is. It's very painful, and it also poses a risk for the personnel who takes the sample, because you are taking sample from 100 people every day, and you get infected, you know, during this sampling procedure. So we thought, if there is virus here, it should be also here in the oropharyngeal region and also in our saliva. So why not take the sample here and concentrate it? So now what we are doing, we are just giving some drinking water to the person and just make a gargle or mouthwash, you know, just mix it in your mouth for 10 seconds and then put it back in a cup again. So even children can do this very easily and give the sample without pain. And since the person give themselves the sample, the person who are taking the sample will not be infected during this procedure. So after taking the sample, we put it in this tube together with our concentrating powder, and we wait only for 5 minutes, and this becomes a gel-like appearance because it will absorb most of the fluid. So the 20 milliliters of this mouthwash fluid now becomes about half milliliters of concentrated solution. Now we take the sample, you can apply directly into a antigen test, rapid antigen test like this, or you can even dip your sample into your gel and see the result. Or we can use this one to do PCR for diagnosis. So we have done a study, a clinical study, comparing, we took samples from volunteers, from nasopharyngeal samples, and at the same time, oropharyngeal mouthwash fluid. And then we did PCR to determine the sensitivity of this technique. So when you look at this sample, it was 363, patients that included in the study, and we did PCR from nasopharyngeal swab, gargle and mouthwash, and concentrated gargle and mouthwash. We have seen that out of 363, we detected COVID-19 in 114 patients, and the detection rate from nasopharyngeal swab was 66.7%. However, when we did concentrated mouthwash, the sensitivity became 88.6. So we can detect even better, with better sensitivity, COVID-19 in mouthwash after concentrating the sample with this polymer that we call MyMagicon. So the advantages are that we are now using a non-invasive procedure. The patient can give itself, and it eliminates the risk for infection for the personnel taking the sample. And instead of taking a very small sample with nasopharyngeal swab, now we are watching the whole mouth and oropharyngeal region with this water and concentrate it to a small volume, and we can do now tests with better sensitivity. Okay, so that's all our contribution to our lab. This is our university here, and this is our research team, some teachers and students. We also have in our university a postgraduate department, which is medical biotechnology, and we have about 80 students right now, and we try to develop this kind of innovative technologies to improve health sciences. Okay. So that's all I want to present. If you have any questions, we can talk about.
There's a question from chat part. Which diagnosis could you suggest for the diagnosis of COVID-19? Which technique could you suggest? Which is the
Yes.
best actually when we consider all the all parameters?
The gold standard method right now is PCR because it has the high sensitivity. However, it takes time. As you know, we get the results one day after we obtain the sample. So if we can use, I believe, these rapid antigen tests together with PCR, in fact, because with rapid antigen test, you will get your result within 15 minutes. So if it is positive, it is very important. It is very precious because you can isolate this person now from the community as soon as possible. If it's negative, but if there are still symptoms, related to the disease, now you can take a sample and do PCR for a test with better sensitivity. Okay.
Okay, thank you. There's another question from the chat part. Is there any other reason why low sensitivity of cassette test, or the only reason is low concentrated sample?
I couldn't understand the question. Can you repeat the question?
Is the only reason that the cassette test has a lower efficiency is not much as effective? Is it only because the
Yes.
lower concentration sample, or are there any other reason?
No, because when you do PCR, you amplify the RNA. So you reach billions of times, more concentrated sample by amplification of RNA. However, in antigen test, there's no amplification step, whatever you have. So you need at least, for example, 10 million virus in 1 milliliter of your sample so that you can detect by antigen. Even when we concentrate our sample, we concentrate about 20 to 40 times with our polymer. So we increase the sensitivity, but still, so we can use our concentration step with PCR or with antigen test also. So we try to approach regular PCR by concentration of the samples with antigen test, of course. But still, we also can increase the sensitivity of PCR by concentration. Okay. So both techniques have their own advantages. One is much more simple, rapid, but PCR is more sensitive, but it requires some trained personnel and it takes some more time to get the result.
Okay, thank you.
Okay.
Can I ask a question?
Yes, of course.
Hocam, when will we be able to use the smartwatch? Because I haven't done any tests for COVID and I'm scared.
Okay. So the approval from our Ministry of Health is done right now. So it's available right now. Now we are waiting for the laboratories to start using it. In fact, it's already been sent to other countries like Holland. They started to use it, but in Turkey, very, very limited number of started just last week, I think.
Very nice. What about the economics? Is the other test cheaper or is this one cheaper? Which one is, or
it
can
doesn't
we say
change
anything like that?
much the expenditure because you have to use a swab and a transport medium when you take your nasopharyngeal sample. So if you take mouthwash, you don't need this.
Yeah.
So these also have a cost. So these together do not, does not change the, cost much.
Okay.
It's about the similar cost,
About the same.
but of course we have much more advantage, in taking the oral sample compared to nasopharyngeal sample.
Okay, thank you. I'm very happy to hear this news. Thank you so
Okay.
much for your talk.
Welcome.
Thank you for speech of Professor Kujigöz. We can continue with the new topic is system biology with Christopher Mayek. Let me give a brief information about him. He completed his doctorate at Colorado State University in biology zoology degree. His areas of interest are system biology, improving honeybee health, animal behavior. Dr. Mayek has many awards as well. I'm giving the floor to Dr. Mayek. Please, Doctor.
Okay, thanks very much for that introduction. Hopefully all of you can hear me okay. My microphone is a bit quiet on this computer, so just...
Yes, Doctor, your voice is coming from deep a little bit.
Okay, good, great. So you can hear me okay, excellent. So thanks very much for the invitation. I really enjoy presenting at these student organized conferences. I find them well organized, and I really enjoy it.
Dr. Maya, can you lean on to the laptop a little bit more?
Sure.
It's very difficult for us to hear you, I guess.
Yeah, the volume. Yeah. Okay.
Yeah.
I'll talk a bit loud, so hopefully it comes through a bit better. Thanks for the invitation, and I really enjoy presenting at these student organized conferences. And I've really enjoyed the first two talks. I've actually had a nasopharyngeal sample done recently, and it is very painful, so I can confirm that. So I look forward to seeing these new technologies and discoveries being developed in Turkey. So I think both have resulted in a positive message for Turkey and an inspirational one. And that's really great to see. I'm going to share my screen now, okay, for my talk, in which we'll switch gears a little bit to focusing on honeybees and honeybee health. And I'm going to show you how a systems biology approach can be used in order to improve honeybee health. So this is an area that I've been fascinated with, and it's something that we're currently pursuing in my research group right now. And here we have a picture of a honeybee, and showing that honeybees here are linked to agricultural production. And with that increased agricultural production, so the increase in crop yields from their pollination services, they aid in putting food on the table. So their health is critical to our well-being because they play a critical role in pollinating many of the fruits and vegetables that we eat on a regular basis. So here you can see that honeybee health is in decline, and it's on a global level. So highlighted in orange are all the countries that have been affected with this most recent decline in bee health. So this started around 2006 and has continued through today, where some of the bee declines have been documented throughout the world. And this is a problem because it's relatively consistent. So we see over the years that the number of losses here are quite high. So they're around 30–40% for a beekeeper. And these losses exceed what is expected by the beekeeper, or what's considered to be acceptable from the beekeeper, which is about 15% loss. So what this is highlighting is that the losses are at an unsustainable rate, and we may be running out of bees, which would be necessary for the pollination services that they provide. So the most recent decline in bee health is what's considered as a complex problem, which means it's multifactorial. So there's a number of factors that are thought to play a role in the most recent decline of bee health. So these include pesticide exposure, new parasites or exotic diseases, poor beekeeping practices, climate change and changes in weather patterns, poor nutrition and lack of foraging habitat available, and then also issues with the queen and poor genetic diversity to be able to adapt to these different stressors. So as I mentioned before, the bees are critical for food production. So this is the most pressing issue because honeybees pollinate about 1/3 of the food that we eat every day. And beyond this, they're also important for a number of products that they produce. So you're familiar with honey that they produce, as well as pollen, propolis, bee venom, royal jelly, and beeswax are all bee products that we also make use of. So if the bees were to die, all the honeybees, this would cost the world about 150 billion euros. And that's about 10% of the current market value of food on the planet. And what's interesting is that Turkey is home to the largest number of beekeepers per capita in the world. So beekeeping and honeybees play a special role in the economy here in Turkey, as well as the culture and the history of Turkey. And so with a systems biology approach here, is a strategy or a way to approach a complex problem in order to understand how the parts of the system are interacting to produce changes in the system. So this schematic here shows you how a systems biology approach is conducted. And typically, but not always, it involves using different omic data sets. So you see this here. And what you see is going across these different omic data sets, the analysis focuses on the interactions across the omics data set. So this fits into the field of big data or big data analysis and starts with these large data sets. And then from here, what's done is usually some data filtering and data organizing and the integration of these data sets. And sometimes these omic data sets can be obtained publicly, so from online sources or databases. And with the integration of them, these different analyses techniques are used. So there's some network-based analyses, multi-step analyses, matrix factorization techniques, as well as Bayesian methods. So these are typical statistical analyses tools that can be used to focus on the interactions themselves that drives the entire system. And in parallel with this, some modeling can be done as well. And from this sets of analyses, the idea is that more specific hypothesis generation can occur, or a model can be developed that can be then validated. And with these new hypotheses and new models that emerge from the big data analysis, you can then further refine your experimental research and test those newly generated hypotheses. So one advantage is that systems biology is considered to be unbiased in the sense that you're allowing the data to drive the hypotheses that are being generated, as opposed to thinking of a hypothesis before doing the experiment in which you may have inherent biases in developing that hypothesis. So this is typically used in the biomedical field, and one of the goals is to come up with new drug targets, biomarkers for diagnostics of complex diseases or disorders. And then the idea here is eventually to get some of the treatments and trials completed and get some of these drugs or biomarkers to be used in the clinical practice. So that's some of the overall goals or outputs of the systems biology analysis. So what we've proposed in my research group in collaboration with Rob Brodrup, Helen White, and Anthony Meshram from the US is using exposomics. So this is measuring the total exposures over the lifetime of an organism and using these to try to predict a phenotype or a change in a phenotype. So in this case, it would be, for us, bee health, or you can consider it as human health. And the idea why we want to use exposomics is it's aiding in characterizing the environment. So we've had a lot of studies that have used transcriptomics or genomics to understand the internal environment or the genes that are perhaps linked to, say, cancer or Alzheimer's or other health disorders. But we know also that the environment influences the whether or not you end up getting these particular diseases or disorders. And so in an effort to try to characterize that environment or those environmental factors, the exposome field has been created and recognized, and it's been in existence for about 10 years now or so. So again, we have, in this case with human health, a number of exposures throughout a lifetime. This could include exposure to pollutants or different dietary habitats. It could be stress from your social environment. And we know that these environmental influences impact the individual, and we can measure the phenotype of that individual, in this case, human health. So with measuring these exposures, there's also changes in the metabolome. So with this analysis technique, the metabolome can be accounted for, and sometimes there's changes in the metabolites that are linked to the exposures, which help aid in predicting changes in the individual phenotype, in this case, health. So our proposal is to use this exposomics within the context of a systems biology approach. And so here, what we see from the biomedical field, typically these are inputs into the analysis. So you may have some experiments with transgenic mice, you may have blood samples, for example, and you may actually have data from clinical studies and gain data from, say, autopsy analyses. So these go into the analysis, these phenotypic measures, and they're usually combined with omics, which help characterize the internal environment of the organism. So we have things like proteomics, metabolomics, transcriptomics, and genomics or epigenomics. And the key for doing systems biology is using a number of bioinformatic and statistical tools. So what we've proposed is to instead apply the systems biology paradigm to looking at the health in bees instead of humans. So we can have healthy and unhealthy hives, and then use these omic tools to characterize the bee itself, the internal environment, and then on top of this, we can add exposomics to help characterize the external environment that they're exposed to, and then be able to look at to see how these interact and hopefully associate particular biomarkers that help predict whether the hive is going to be healthy or unhealthy. So from this research, we may be able to develop novel biomarkers or even a biomarker pattern. I'll show you this. We can also gain insights into the pathogenic effects from an infection on a molecular level, and focus on biochemical pathways such as metabolic pathways that might be altered from a number of infections or exposures to stressors. And then eventually, hopefully, develop novel therapeutic targets for new treatments to improve bee health. So here's showing you an example of a workflow where we're sampling forager bees from the hive, and these are divided into a subsample in which half of it can be used for molecular disease screening. So we use PCR and qPCR techniques for that. So you just saw that in the previous talk and learned about that. And again, it's very sensitive, so we can pick up whether or not the organism or the honeybee is infected and in the abundance of infection. And then we can couple this with the exposomics. So looking at the exposures, and we use gas chromatography quantitative time of flight analysis for this, which is high resolution mass spectrometry, to be able to identify a number of exposures that based on their chemical properties are identified from an online database. And we then integrate those and use Mass Profiler Professional as a software, or you can use Python or R to do the statistical analyses and be able to integrate these data sets. So then focusing on the interactions from these two main data sets, we can then start to see how the exposures might lead to infections of diseases, so being particularly susceptible to the a particular disease such as Nosema cerana, which is a fungal pathogen. And then also we can do what's known as KEGG pathway analysis and look at the metabolic pathways and see how certain metabolites might be increased or decreased in relative abundance, which might give insights on how the physiology of the bee is being altered from an infection. So a master student in my lab is responsible for doing the most recent systems biology analysis. And here's an example of the workflow of which he's created and has gone through in order to identify biomarker profiles that can be used. These chemical biomarkers from the exposures can be used to predict whether or not a hive will be infected with a particular disease. So this involved sampling beehives. We had sampled 29 hives 3 different times. We combined this data, and again, half the samples were used for GCMS analysis, and then the other half of the subsamples were used for molecular disease screening. We combined that data, it was filtered, normalized, and then we classified the hives whether they were infected or non-infected based on their disease abundances. And here is showing you all those different diseases that we analyzed, which are all the most common diseases that are linked to the most recent bee declines. And then from that, we are able to extract out features using a PLS analysis. So this is a multivariate analysis tool, which then allows us to extract out the particular chemicals that are most highly associated with those diseases. And then as a last step, we used machine learning to validate, using algorithms, we validated the chemical biomarkers to see how accurate they are in predicting a particular disease. So from this, we could then select the best performing model based on the ones, or based on the features extracted that had the highest level of predictability based on the machine learning. So here's an example of some of the results that we've obtained. And here we have different diseases. So this represents varroa mites, varroa mite count. So this is a mite that impacts the bee health quite a bit. We have Nosema cerana, which is a fungal pathogen. And then we have European foul brood, which is a bacterial pathogen. So what you can see from this Venn diagram is we have chemical biomarkers that are unique to each pathogen. And then we have some that are common to all the pathogens. So this is relatively powerful because we have now chemical biomarkers that we can use to diagnose if the hive is just infected or not with any 3 of these, or we can have biochemical or chemical biomarkers that are specific to each disease as well. And here's an example with the viruses, some of the different viruses that infect bees. And you see a similar trend where we have some that are common to all of the bee diseases. And then we have some, or quite a few, that are specific to each virus as well. And so this is the result summary here showing the 5-fold cross-validation using the machine learning algorithm. So this is using the SVM method. And what you see here is what you generally hope to see is that this curve is deviating quickly from the y=x line. So this means that the accuracy is relatively high. So we have the area under the curve value, which represents the accuracy. And here we have 95% accuracy, for example, predicting sac brood virus with the chemical biomarker profile that we've developed for this, or selected for predicting this particular infection. So you see that consistently throughout. There is some variation. So we have more difficulty, for example, predicting acute bee paralysis virus, for example, this complex. But in general, we have relatively high values. And overall, after doing a number of validation methods, we find on average, for all the diseases, we have about a 90% accuracy. So we have about 90% confidence in using these chemical biomarker profiles in predicting if the hive will be infected with this disease. So these are some examples here of the top hits that we found that are associated with different bee diseases that we screened for. And what I want to point out here is most of the chemical biomarkers have not been associated with bee health before. So we're pretty excited about this result because there hasn't been any previous link to these exposures and the fact that they become infected with these particular diseases found in the literature before. So it's a completely new result. And what we found here is with another study is we can actually capture these chemical biomarkers without being very invasive. So you saw that again in the previous talk, how if the technique is more invasive, it's generally not a good thing. And so here we've done a study where you can place silicone wristbands, and we've actually been able to absorb some of these chemical biomarkers and pick these up with GC-MS analysis later on in the laboratory. So what's key to this is it's very easy for the beekeeper to do. They can place these in the hive without opening up the hive. And also this involves no need to sacrifice any bees in order to screen for the pathogen. So it helps the beehives as well, which is normally required in order to see if a beehive is infected or not. So the idea here is to have the beekeepers be able to use the silicone bands, and then the Turkish beekeeper will then submit these hopefully to a centralized bee health center, ideally, in which a high throughput analysis that can be used, such as GC-MS, can be used to look for these chemical biomarkers and then rapidly tell whether or not the hive is infected to the beekeeper. So this is the main issue. So same with COVID, where you need rapid diagnostic tests. The same is true for these bee diseases, which currently doesn't exist. So that's the overall goal or aim with this research. And then in addition to this, recently applied to an EU grant to be able to use bees to monitor the environment and to determine whether using these new farming techniques as part of the EU Green Deal will actually result in lower exposure to chemicals and pesticides and pollutants to bees. And so this systems biology approach will be combined with these IoT sensors that continuously monitor the internal hive. And with this combination, we could use bees as a continuous monitoring program to determine how good these new farming practices may be in terms of reducing pesticide exposure to bees. So with that, a small plug to graduate students. Feel free to apply to Sabancı. We'd love to have you. There's a number of benefits, and I'm interested in taking on students this upcoming school year. And there's other new faculty joining that would be happy to host you. I've had a number of students in the past and hosted also a number of undergraduate students with our peer research program. So keep us in mind, okay? And with that, I thank my collaborators that the collaboration initiated at Swarthmore College in the US, and then also these students here who've helped me with the work that you've seen today at Sabancı University. I also thank you for your attention and be happy to take any questions.
Thank you, Doctor. Doctor, actually, I have a question, but I think not related with exactly this topic, but again, about the animal behavior, especially about the bees. Did you observe any changing about the behavior of bees? Did you observe? Because as you know, there is many conditions like climate condition, like ecologic and pesticides, etc. Did you observe any changing about the behavior of bees? Thank you.
Mm-hmm. Yeah, so there's been some studies done, and some studies I've done as well. So if we focus on one particular pathogen? Well, there's general symptoms that you'll see if they're infected. Sometimes they're hard to detect, but bees will generally, if they have viral infections, sometimes they'll twitch or be immobile outside the hive. And also with fungal pathogens, they can change their social behavior sometimes. So they can detect infected individuals and they will either, sometimes they'll kill this individual, sometimes they'll tend to it and care for it and give it extra food. And other times they'll avoid it completely. They do this which is thought to help reduce opportunities for disease transmission. So with these individual factors, sometimes we can see certain changes in behavior, either at the individual or social level. And there are particular signs that the beekeeper uses. So for example, if the bees have been exposed to pesticides recently, a lot of times you'll see a large amount of dead bees out in front of the hive. And that's a classic symptom of bees being exposed from aerial spraying of pesticides. Yeah.
Thank you, Doctor. It was very informative speech. I think we can continue with another speaker if you haven't got any question, because I couldn't see any question on the chat part. Thank you.
Yeah, not a problem. Thanks very much for your time. Yeah.
Thank you.
Thank you, Doctor. It was very nice to listen to you.
Thanks very much. Thanks very much.
We will continue with really interesting topics about microbiota with Professor Metehan Özen. The presentation name is Miracle of Microbiota. I will give brief information again, then we will start the presentation. Professor Özen completed his specialty in pediatric in Cerrahpaşa University, Faculty of Medicine. During the years, he focused on human microbiome studies. He provided the first probiotic book to be published in Turkey. He organized the world's first pediatric meetings, which name is International Symposium of Probiotics in Pediatric. And he's currently studying in Acıbadem University Hospital now. And we are with Doctor. Please, Doctor, we are listening to you.
Thank you very much. Thanks again for the invitation. It's really nice to be with the young people.
Thank you, John.
I also listened to previous topics. They were quite interesting, really. And also Professor Tanıl, we are working together in Acıbadem. I'm a clinician, so my topic is generally will concentrate on some daily subjects, daily problems. I want to talk about microbiota, but it's a very huge and wide topic. So I wanted to focus on maybe upper respiratory tract infection, because it is the #1 reason all over the world. This is the #1 reason to go and see a physician. And also antibiotic associated diarrhea, AAD. When you use antibiotics, unfortunately, you may face some side effects like diarrhea is the simple one. And as you will see in the coming slides, we are the #1 country in the world, one of the best countries who uses most antibiotics. Of course, it is not a very good choice to use more antibiotics, but unfortunately we are. My presentation will focus on the increasing number of studies and the global burden of respiratory infections. It causes the antibiotic overuse, and there is a global burden on it. But there is another phase of the problem. There is a personal burden. And since we use so much antibiotics for upper respiratory tract infections, can we prevent, can we diminish the number of infections and maybe the use of antibiotics, of course? Nearly 10 years ago, I was the editor of the first journal. It was the Probiotic and Prebiotic Chronicles in Turkish. And the cover was "The Good Men Are Working," so be at ease. And the first editorial article, I summarize all the studies from 2000 to 2010. In the last decade, I said that the numbers are increasing so much. It's more than 100 articles in a year. It is fantastic. Now I'm laughing because now you see the numbers. As you see, it's more than 1,000 in a year. So the last decade saw uprising like a rocket rise, as you see, the microbiome, but it is not only the gut microbiome, because the first studies concentrated on gut because we think that it was they all live in our gastrointestinal system, but we know that they are everywhere. Even in our brain, there are some particles of them. When you see the red zone study, the first studies was about the acute diarrhea because gastrointestinal infections. But now we know that they are quite important. I will talk about the balance and the dysbiosis. So they are responsible in the pathophysiology of many events and many diseases. Now we have studying for the development, nutrition, against allergy. 10 years ago, when I talk about that, we will see bacteria. Probiotics are the live microorganisms, the bacteria or some fungi. They will be available in the growth formula for the infants, for the babies. Everybody was laughing at me. Now in Turkey and in the world, there are only 1 or 2 growth formula remaining without bacteria. Now all the formulas are containing bacteria because we know that the breast milk, the mother milk, is containing more than 100 bacteria of the mother. These are all new information coming in the last decade because of the technology, especially the PCR technology we have. Instead of culturing them, now we are detecting the proteins, the particles of the microorganisms because of the sophisticated techniques. And we now know that even the breast milk contains more than 100 bacteria. This is a new study, a new article. As you see here, the microbiome, the human studies, now there are more than 16,000 articles now in the last decade. And it is not only the microbiome or metagenomics, not the human, it is also all the living animals, such as agricultural crops, the forest trees. Now the science is searching for the microorganisms available everywhere. 10 years ago, there were only a few journals publishing these studies. Now, all the best journals like Nature's, New England Journal of Medicine, Lancet, in every issue, you can see a study about the microorganisms or probiotics. And there are now some reports, committee reports, national study teams, books, and there are websites. And as you see now, only the problem is we have some commensal bacteria, fungi, virus living with us and good for us. And there are also some pathogens. They are harmful, but there is a balance. So when you trigger the factors which causes imbalance, we call it dysbiosis. This is the biggest problem now for the humankind in terms of health. Most of the diseases now we have, you will see here, because of dysbiosis. 10 years ago, a group from Dallas showed that the people living in the same geography, same age, same sex, sometimes are siblings. When you check the healthy control, these are the phyla of the bacteria. There are 6 major phylums, but more than 10,000 bacteria and viruses in the human being. When you see the phylums, you see when you compare the healthy control and the patients in the inflammatory bowel disease, which is quite common in our population also. In type 2 diabetes, we are the #1 country. This is a kind of simply obesity, let's say. So we are one of the best countries in terms of obesity in the world because we have a very good cuisine and we eat so much. You see that the proportion of the phylum, the families, the bacteria are different from the patient. So this was a pioneering study. When a doctor or a researcher read about this study, they wanted to study all the chronic disease they follow up. And 5 years later, you see Hashimoto thyroiditis, asthma, eczema, psoriasis, which are now quite common in all over the world, also in our population, in our country. In the pathophysiology, there is a dysbiosis. When we come to these days, there are more than 150 diseases. We nearly studied 170. I mean, 90% of the chronic, allergic, and some autoimmune diseases, when we check for the risk factors or etiology, or we call it pathophysiology, now we see that there is dysbiosis. And we have, when you have some disease like this, chronic disease or autoimmune disorders, you go and treat it with drugs, and your appetite is not good. You take so many drugs. So all these factors are also impacting your health, and it's causing again a dysbiosis. Now you have a vicious circle, and your health is going worse and worse. So when we understand that the dysbiosis causes many diseases, the problem is, can we prevent or can we treat it? It was very easy to prove it when you are talking about acute infections diarrhea, because you have bad microbes, and when you ingest good microbes, they fight each other in the intestine, and you have resolution. But it was difficult to prove it in the autoimmune disease, allergic disease, or disease infections of the respiratory system. So it doesn't make sense in the beginning. You will ingest something, and it will be good for your nose or your pharynx, let's say, or your lung infection. But we understand the etiopathogenesis. So your immune system is 70 or 80% is located in your mucosa, in your gastrointestinal systems. And when you receive some beneficial bacteria, they boost, they talk with your immune system, they stimulate them to fight more, to produce more anti-inflammatory products. So now we know that when you see these bold ones, the bold written ones, these are many, there are many studies proving that they are quite effective, but this is an old study from 6 years. Now colic is also in bold, and common infection disease, including upper respiratory tract infections, which I study in our country, in our group. Now we prove that they are effective against the, in the prevention, not treatment. It is quite different. We treat gastrointestinal infections, but we try to prevent upper respiratory tract infections. It means that you have to use for a long period. And we are talking about this, we are organizing not only the National Congress, also we are this year, hopefully we will prevent the 7th International Congress in Spain. I'm not sure if I will go. We are organizing with my partner, Dr. Enar from Eskişehir, and some European partners. But it will take 3 or 4 days to discuss all of this. But I have only 20 minutes to tell you about all this. And God help me. I want to concentrate on the respiratory tract infections, because the viral infections are the most common reason to go to a doctor. And influenza is the most trouble virus, because it causes most hospitalizations and unfortunately some deaths. As you see, especially over the age of 65 and chronic people, it causes death. We used to tell about this in the lectures, but this year, you know, the COVID, because of COVID, is changing. Now the influenza is here. This is the fatality rate, and this is the COVID fatality rate. As you see, it is 100 times higher than the influenza mortality rate. But you see, as you see in the table, we have more viruses like Ebola or Hendra, and their fatality rate is unbelievable. So let's pray that we keep safe from these viruses. Even the coronavirus, now it is the fatality rate is something like 1% among these days, but it is still high. But I will talk about this non-influenza respiratory tract infection. We have more than 100 viruses. They cause the most common infection. It is the #1 reason to spend more money. There are some indirect costs, because when your child is sick, you cannot go to work or your school. So you have to pay more, or you have to refrain from the work. But my problem as an infection disease doctor, besides the cost, there are some adverse effects of antibiotics. It causes dysbiosis. Unfortunately, it causes antibiotic resistance. I'm an infection disease specialist. I use antibiotics so much for the people in the intensive care units. But from the outpatient clinic, I mean from the patient who have only viral upper respiratory tract infection, we know that nearly 90 to 95% of the upper respiratory tract infections are viral. But unfortunately, we use more antibiotics. And WHO announces that even the minimal reduction in the use of antibiotics has paramount importance. Only 10% decrease will be a salvage for the humankind because the numbers are really frightening. I will share you the numbers. This is the best institution in the world to follow the infections. As you see, even in America, the inappropriate use, I mean the acute respiratory, the viral use, as you see, half of the antibiotic use are misuse. This is the Canada where they look after less numbers of patients, because when you take care less number of patients, you can concentrate much more and you can talk with your patient and maybe you can convince them this is a viral infection, you don't have to use antibiotic, because the patients always want more antibiotics. Even in Canada, as you see, for non-bacterial acute upper respiratory infection, 40% of the patients receives antibiotics. This is a huge number. And when you see the OECD countries, of course, mashallah, we are number one, using more antibiotics, more than anybody, any country in the world. When you check for the antibiotic resistance, I mean the bacteria who are resistant to antibiotics, we are number second after Greece, but don't be sad about it. So in the future days, we will be number one. I'm always talking about this in my lectures. And I always talk that in 2023 is our big dream. We will be the number one in the resistance. But anyway, it is 2020. Last year, we were past the Greece. So we are number one in the Europe. But I didn't know that India, China, Russia were worse than us. So this is a study from Koç University, Professor Önder. So I am really disappointed to see that we are not number one in the world. We are number one in the Europe. But there are some worse countries, as you see, India, China, Russia, Romania. This shows the antibiotic resistance, my friends, and it is really, we have to cry for it because they kill people. Thanks to the very good actions by our Ministry of Health and because of the advertisement and because you cannot buy antibiotics from pharmacy now without prescription and some issues like this. The antibiotic use is decreasing in our country, but even in this decreased form, we are number one in Europe. As you said, now there are some many action plans. It's coming from European CDC. It shows that in Europe now, last year, in Italy, more than 10,000 people die because of the infections which are antibiotic resistant. There are not enough antibiotics to cure them. So this is a very sad number, 10,000 patients because of untreatable infection. So when you multiply, we estimate that it is nearly 30,000 people in our country dying from because of antibiotic resistant antibiotics. Now everybody is afraid of COVID or some other thing, but in the normal time, in the normal years, we already lose nearly 30 to 40,000 people because of the infections which are resistant to antibiotics available in the market. So this is a very sad situation. It is not, the Europe is in the best condition, as you see. When you check the Asia or Africa, you see the numbers are very huge. And the World Health Organization announces that if we do not find any new antibiotics, your first presentation, the Professor Dr. Hassan talked about the new drugs, better drugs. Inshallah, they find something new, because if nobody finds anything new, they assume that it will be the number one reason. So we will go back to the Stone Age. Before the penicillin, as you say, it will be antimicrobial resistance will cost 10 million lives, whereas the cancer will cause 8 million. So this will be a problem before the penicillin area. So we are going to the Stone Age. So we should find new drugs. And until we find something new, we should use less antibiotics. If it is unnecessary, which is unfortunately 70 or 80% unnecessary, we should diminish the use of antibiotics. As you see, the World Health Organization is announcing it. The world is running out of antibiotics. This is now global threat. This is #1 threat for the humankind now. And this is the second problem. The first problem is the increasing antibiotic resistance, and we have no new drugs. The second problem is 20 years ago, we realized that your body is a planet, but it is a complete human when you have microbes. So we are not alone. We are not alone in this space, but we are not alone at home. As you see, we have an invisible microbiome, and their number is higher than our cellular number. And as you see, what causes dysbiosis, the factors impacting your microbiome, it's not only pharmaceutical antibiotics, it is because of the wrong diet, unfortunately, because of stress, which is plenty in our country and in Istanbul, because of birthing process. Unfortunately, more than half of the babies now having cesarean section, which it must be most 10%, but it is more than 50%, and this impacts the microbiome and immunity in the birthing process. And also, instead of breast milk, unfortunately, 25% using formulas. Of course, we should use, but when necessary. We should not use them when the cesarean section is necessary, but only in 5 to 10%. But nowadays, more than 50%. All the ladies do not want to experience so much pain in a long time. So it's a very easy, only half an hour. And geography, all these factors causing dysbiosis. It is a very long process. It takes our 2 days to talk in our congresses. But when you have dysbiosis, the inflammatory cytokines increase in your body, and because of the blood or the lymph, they go to the target organs. Whichever organs they visit, they causes inflammation, and inflammation causes autoimmune disease. This is shown in more than 150 diseases now, autoimmune disease, some cancer disease, most of the allergic disease, and so on. So now this is the problem. So if you are not live like your fathers, mothers, if you are not eating like them, you're using so much pharmaceuticals, thanks God we have plenty stress, and instead of vaginal delivery, we have cesarean section. So now most of the patients visiting in our hospitals are because of these autoimmune disorders. Because we knew that when you use antibiotics, we didn't know that the dysbiosis will last only 1 month. 10 years ago, we believed that it lasts 1 month, but now the study is continuing, and we see that even after 4 years, the effects are continuing, and the studies are showing very well that when you are pregnant, if you are using antibiotic, it may increase. Please do not misunderstand me. I do not claim that it causes. It increases the risk of having some asthma, allergy, type 1 diabetes, and this loss of microbial diversity and enrichment may cause some chronic diseases in both for you and both your children. So these are very long subjects, but I want to show you one example, because 5 years ago, I was somewhere in the Eastern Turkey. I was talking about the dysbiosis, and some rheumatology professor told me that no, there is no any link between rheumatoid arthritis in the elderly people and in between the dysbiosis, which is caused by antibiotic. So this is a very new study. Amazing number of rheumatoid arthritis patients, more than 22,000. It's an amazing number. It is matched 1 to 4. So there are 90,000 controls, and they follow up them 10 years. It's a huge number. We cannot organize such big studies in our country. So this is a very group study. It takes 10 years to publish it. We are not such patient beings in our country. But when they check it, they see that you see the crude odds ratio. It shows your risk. When you use 1 box of antibiotics or 4 boxes of antibiotics in 10 years, it shows the risk of having rheumatoid arthritis. So it increases with every package you use. So you are sick, you have an infection, so you have to use. Of course, we have to use antibiotics when necessary. Then we should do something else. What can it be? We always think that clindamycin or anaerobic antibiotics cause this, but even the antibiotics like macrolides, which have little impact on microbiota, or we believe that there is a little impact on microbiota, we now see that many of the antibiotics, even only the simple penicillin, the first antibiotic, has profound impact on microbiota. So what can we do? This may be an option. Another option is having a healthy diet, to give up using the Western diet with high fat content. This is the longest paper written in the literature. We wrote it with McFarland, and it shows that some probiotics, not all of them, you see the line here, vertical line. So these are the beneficial effects of some probiotics. So when you use probiotics together with antibiotics, it shows that it decreases your diarrhea, it decreases your Clostridium difficile, which is a marker for showing the dysbiosis, let's say, in basic terms. It diminishes your risk. So this may be one option, to use probiotics when using antibiotics, to use more fermented products like yogurt, kefir, and some fermented products are very beneficial to fight against dysbiosis. So the beneficial effect may be both ways. One way is, this is when you do not use probiotic. This is the effect of antibiotic on your microbiota. This is the depth of the dysbiosis. So when you use probiotic, it may be either diminish the effect, impact of the antibiotic, or lessen, shorten the duration of the dysbiosis. So this both effects, methods may be effective. It's not proven yet, but these are all hypothetical ways. But anyway, it shows some protection against Clostridium difficile and prevention against antibiotic-associated diarrhea. So there are some beneficial effects. The second method, you should diminish, you could increase, support your immune system and diminish your indirect way, diminish your infections. When you check Google, if you write how to support immune systems, less than 1 second, there are 200 million results. So we are not the only country on the world to eat which herb is available, because when you check the internet, which herb, which plant do you eat, they are beneficial. No, we do not act like this, because there are also harmful effects of plants. You should study, you should make some double-blind study to show. As you see in a website, whatever you eat, whatever you use, they have immune boosting effect. No, this is not true. You should prove it. So this is the first paper to show the beneficial effects of some beneficial bacteria for the prevention of pediatric upper respiratory tract infection. This was the first review. These are the tables of the detailed tables of the study. These are the results. There were high quality 14 double-blind randomized placebo-controlled trials. There were nearly 6,000 children, and most of them before the age of primary school. I'm in the daycare centers. As you see, in some of the studies, 12 of the 14 studies, they reported the incidence of infection and severity of the symptoms. They showed that 7 had significant decrease in the number of infection. Even if you have infection, your symptoms are decreasing. So you have mild infection. This shows us, this guarantees that you use less drugs and antibiotics. So not every probiotic is probiotic, beware. Not every product available on the market is not effective against infections. So if you know the literature, if you go and ask the right doctor and the pharmaceutical pharmacist, they will tell you which probiotics are best to prevent, as you say, infections. And also some of the studies, they show that antibiotic uses decrease. So we updated this article last year because there were amazing number of studies increased. Now 33 studies, more than 11,000 children. Again, in 20 of the 30 studies who reported the incidence, the number of infection, there is decrease. So you will know which probiotic is good against the respiratory. So every probiotic is different for gastrointestinal infection, allergy, nutrition. So this is a very huge topic. That's why the Congress is lasting 4 full days. So every bacteria is effective against some other, not every bacteria, sorry. Some bacteria which have clinical studies, then it is called probiotic. If you do not prove it with clinical study, it is only bacteria. If you've done a human study performed, and if you prove that it has some beneficial effects, then it is called probiotic, but it is not common in the market. So whoever sells some bacteria, they call it probiotic. So no people just search for it. Everybody believes that every probiotic bacteria is effective on the market, but it is not, as you see. Some of them are quite effective against prevention of respiratory tract infection, and also it decreases the number of used antibiotics. So it is beneficial, and the effect is more than 25 to 30%. Please remember that the World Health Organization announced that 10% decrease of antibiotic use has a great importance for the human race. So using the correct probiotic, so we used all these studies for 3 months, for a long time, because this is a prevention study. To make it simple, when it is winter in Istanbul, like October, November, when the schools are opened, when the children come together, we start using correct probiotics for 3 months. And I asked the family, was it useful? Was it beneficial for your children? If they say yes, our children has suffered less infections, less antibiotics, then I recommend them to use 3 more months until the April and May, until the summer. So we use it for 6 months. But not every bacteria is good for every people. So it is a complex situation. These are not drugs. So the last slide is, can we save while spending? There is an interesting study in the swine flu outbreak pandemic 12 years ago in Paris. They calculated that if they use probiotic, the correct probiotic, everybody in Paris, will it be cost effective? Because there are only 3 options for cost effective in the medicine. The first one, the great one, is the supplying clean water. This is the most important issue on the world for humankind. The second one, vaccination. As you see, spending money on vaccination saves lives, money, and people. And the third one, online shopping. My wife always claims that online shopping is very cost effective. Of course, I am joking. The third one is maybe using probiotics, because when you use the correct probiotic, as you see in this study, if they give probiotic everybody in Paris, they show that they would save 2 and 4 million infection days, and they will see that nearly 300,000 antibiotic used less. So they would save nearly 15 million euros saved if everybody used probiotics. I mean the correct probiotics in Paris. So to make it short, to take a messages, viral respiratory tract infection is the #1 reason to go to physician. But unfortunately, it has the high mortality and morbidity. So this slide was before COVID. Now after COVID, it was writing morbidity here. Now I added mortality, unfortunately. Antibiotic resistance, it is the major problem for all humankind. And antibiotic-related dysbiosis is the major problem for your individual. Probiotics may decrease, not every probiotic, not in every human, because if you have a very good microbiota, if you have no dysbiosis, then you will have no beneficial activity from probiotics. Certain bacterial strains, certain probiotics looks promising now in reducing the number of infections and severity of infections. But of course, we need more multi-center studies searching for similar outcomes in similar ages and with some underlying disease. And the topic goes on and on like this. So if you want to join, or I also want to invite you to our national congresses. And if you have any further questions, you can email me anytime. Thank you for your patience.
Thank you, Doctor. If you have any question, you can write on the chat part. Doctor, actually, I have a question. If you have any time, may I ask you?
I have a few more minutes.
Okay. I have 4 questions.
The patients are waiting for me. You know, the program is late.
Okay. I will be.
Yeah. Please ask the most important one. Email the others.
Okay. The most important for me, baby food. I mean mamalar.
Okay,
I
formula, we call it infant formula.
Actually, my question about I wrote an article about the baby foods, and there are some side effects with baby foods because they are collecting in the foods raw metals, ağır metaller birikiyorlar. Does it cause the dysbiosis, and what can we do? And the most important one also microbiome therapies. I remember you mentioned in your congress about microbiome therapies. What do you think about these therapies in the Turkey especially? Can we be successful about these microbiome therapies? What do you think?
Your last question, microbiome therapy is a very complex, very problematic issue. So we need more studies, especially the safety studies. There are very useful areas, useful subjects like sometimes we face bloody diarrhea after antibiotic use, and if it is a severe condition, you may also lose the patient. So we should save it for these life-saving options. But it became very popular. They use for everything like asthma, allergy, losing weight, or for the diet options. So this is not approved yet first, but for some critical conditions, we are also using in our country, we are using. And your first question, some foods are containing additives, high metals, baby foods, but everything is containing them, not only the baby, also the elderly food, everything we eat is composed of these high metals or additives, unfortunately. Of course, they are causing dysbiosis. There was a fantastic study also from my partner from Eskişehir. They searched for the heavy metals in the breast milk, the mother milk. And they found some heavy metals in the breast milk. It was very surprising, shocking. So whatever you eat in this country, in this world, unfortunately polluted with high metals. I also remember reading an article about honeybee, coming from the Karadeniz mountains, from top of the mountains. They studied the honey and they found also heavy metals. They wrote, in the conclusion, they wrote that because of the rain is coming from the Karadeniz, Black Sea, and it's raining and maybe probably bringing some pollution like that. Unfortunately, we are polluted. The world is polluted. But most of the formulas now preparing with very high sophisticated techniques in European countries, of course. Not every country is very cautious about this, but some countries, especially the European countries like Germany, France, there are very strict rules. They are very careful about it. They are trying to make it natural or organic, but maybe, of course, they may contain some pollutants, unfortunately.
Thank you, doctor. It was very informative and inspirational for us.
Thank you. There's another question. Does Hashimoto disease has a treatment, or the patients must use the drugs for their whole lives? From Orcha.
I'm not following or treating Hashimoto people. It is another specialty. But if you have Hashimoto, there are some treatment options. And if it goes well, then you do not have to use drug for lifelong. But we know that Hashimoto is very common, especially in Istanbul population, nearly 1 to 5 to 6 ladies in Istanbul having Hashimoto. And we now believe that it is because of the stress, it is because of the diet you consume, and because of the drugs, it's causing autoimmune disease. And Hashimoto is one of the most common in populated countries, and especially ladies, and of course men.
Thank you, doctor, for your time.
Okay. Have a good conference, and I congratulate you so much for this nice organization.
Thank you, doctor.
Thank you.
Can we give a break, or can we continue? What do you think, dear participants? Hocam.
Actually, if you're asking me, I don't have so much time. So if you can keep on doing this, I'll be very appreciated with that.
Okay, doctor.
Thank you.
So I think we should continue with Dr. Ayca Zeynep İlter. The new presentation topic is CRISPR gene editing and its application. Dr. İlter completed her master degree at Yeditepe University in biotechnology degree. And again, she completed her doctorate in biotechnology field. During her undergraduate, graduate, and doctorate studies, she conducted research at institutions such as Berlin Şarait University and Istanbul Aziz Sancar Experimental Medicine Research Institute. And gene
Şeyma, you are breaking up, I think.
Şeyma'cığım, I think there's a problem in your connection, but it's fine. Thank you for your introduction. I can go on from there. Hello everyone, I'm Dr. Ayca İlter. So for now I'm an assistant professor at İstinye University Faculty of Medicine at Histology and Embryology Department, and I'm working with Professor Dr. Erdal Karaöz. And also I have my own technology firm. I'm going to talk about virus and biotechnology, and today I'm going to talk about CRISPR and its applications. Especially I'm going to focus on the cancer biology. That's about the topic that you give me. And I think you can see my slides, yes. And also thank you for inviting me to this wonderful seminar series. So if you have any questions, I'd like to answer them at the end of my presentation. So I know that this CRISPR gene editing is not new for you guys, because there are several research groups that has been conducting CRISPR researchers at Üsküdar University, and I know them very well. So I just want to go into focus on what can we do on the cancer biology by using this CRISPR technology. So basically, what is cancer? We know that these normal cells, they're dividing normally, and if there is anything happens while they're dividing, like they damage, they can be repaired or they go under apoptosis. But when we talk about very basically about cancer development, we know that these normal cells accumulate some mutations, and the accumulation of these mutations drives those cells into an uncontrolled growth. And these accumulation at the end, you can see we have a genetically heterogeneous cancer cell population, and these cancer cells, like I told you, they're uncontrollable, they're growing so fast, and we still have very big challenges in the cancer treatment as well. So this genetically heterogeneous population of cancer cells, they go under metastasis. Like, as you can see, this is a primary tumor, and after that, this tumor just invade through this vascularization system, and we know that they're working with this blood circulation very well. And while doing this, they're using lots of different mechanisms, like extracellular matrix dependent, like enzymes, and they're overexpressing those enzymes in order to invade through this vascularization system. And after that, they're getting into the blood circulation, and then somehow they can survive in the circulation, because we know that we have normally immune systems, right? And this immune system cell can go and detect those cancer cells, or the transformed cells, or like foreign bacteria, but somehow these cancer cells are surviving. And after they survive, they can go to the distant places. They can extravase from this blood circulation, and then they can go to a foreign microenvironment and survive in there. That's what we call the metastasis. as you can see from here, for example, if you have if a patient have a colorectal cancer, it can make metastasis very easily to the liver and the other parts of your body as well. And it's there are lots of treatments about this cancer therapy, but today I just would like to remind you the hallmarks of cancer and how can we compete, or how can we add new therapies for the cancer treatment. As you know, guys, for the who are working with cancer biology, this is like a book for us, the hallmarks of cancer. It's firstly, it was a review, and it's first written by Hanahan at 2000, and after that it was revised at like 2011 because the hallmarks of cancers are just evolving, and it's amazing. It has like 51,000 citation it takes, and still we are citing this paper because it's like the book of the cancer biology, and you can easily understand why these cancers or how these cancer cells can survive. So we know that from the cancer biology, these cancer cells, like I told you, normally our immune system should detect those cancer cells while they're growing, but somehow these cancer cells can avoid from this immune destruct and immune cells, or they can the other one is the tumor promoting inflammation. What does it mean in our system? Sometimes, not sometimes, during this metastasis, the tumor cells can be promoted by immune system cells, and accumulation of mutations can lead to the genetic instability. Or we know from the cancer cell, is anyone working at the labs, I don't know, but they have like immortality. They have a very huge replicative capacity. They're resisting the cell death, and they can activate this invasion and metastasis, like I told you, by using this like matrix metalloproteinase. They can degrade the extracellular matrix beneath them in order to go through this blood circulation system in order to reach them. And the angiogenesis is the other hallmarks of the cancer, because through this angiogenesis, we know that they can go to the distant metastatic sites. They can evade from the growth suppressors, they can affect the cell cycle mechanisms, and also they can deregulate the cellular energetics. We all know these processes, and also I would like to mention that each one of this hallmark is another field. Some of the people in cancer biology, they're working with the genome instability and mutation like several years. Some of them are working with the resisting cell death, like autophagy or apoptosis. So these are very big fields in the cancer research. So I just want to make a small review of them in order to remind you what is cancer and why do we need new therapeutic options for the cancer treatment. For now, we know that mostly, we have like hormonal therapy, for example, for the breast cancer. If it's related with the estrogen, we need a hormonal therapy, and estrogen-dependent cancer cells can be cured by the hormonal therapies. Or mostly we have the surgery, but that during the surgery, because of this, the cancer cells are very like microstructures, but the surgery is a kind of a micro application. So when they're doing this surgery, the doctors doing this surgery, they can spread some of the cancer cell or resistant cancer stem cells around the body, and that can go to that can lead to another relapse in a patient as well. We know that for the blood cancers, we have bone marrow transplantation. We know the chemotherapy, but the chemotherapy, as you know, it's not a targeted therapy, but it could be. Also, people are also evolving this chemotherapy. They're developing it because we know that, and when we compare to the other therapies, chemotherapy is the cheapest one just for now. For example, for the personalized therapy, the costs are too much. We have radiotherapy for other type of cancers, and we have the immune therapy. It's very new. Actually, we accumulate some of the data about the immune system and cancer cell interactions. So according to this, people are just developing new immune therapy methods in order to cure cancer by people's own immune system. So you remember, this is the hallmarks of cancer, and for each one of the hallmark of the cancer, CRISPR can find some new treatments or can add new treatments to the cancer therapy. For example, for the sustaining proliferative signaling, you can model and activate oncogenic mutations to avoiding growth suppressors. We know that they're epigenetically silenced tumor suppressors, so we can change this the epigenetic silencing mechanism of that. Avoiding immune destruction, I'm going to mention about it. They're very recent immune therapy method. We call it CAR T cell therapy. So what people are doing, it's like an ex vivo therapy. The people, the doctors are taking your own T cells because somehow your T cells or cytotoxic T cells cannot recognize those cancer cells. And they're educating those T cells at in vitro at the labs. And after that, they're giving those T cells to the patients in order to find, in order to catch the cancer cells and destroy them. And we know that the telomere length is another problem in the cancer cells. So, and also the epigenetic editing to repressing the cytokine repressors. Mostly for the CRISPR, people are using the screening for candidate metastatic genes. And for the drug-resistant genes, we can find drug-resistant genes by using the screening technology as well. And not only the, I'm talking about the exon of the genome, but also for the microRNAs, there are new technologies are developing in order to detect the or change the microRNAs. Also, you can target viral genes that inhibit the apoptosis as well. And there are lots of other applications, of course, but it's a very nice, how can I say, review that shows you that gene editing or CRISPR is involved in our system so much. So we can find a new cure, new therapeutic strategies by using CRISPR-Cas9 or CRISPR technology in cancer therapy. So most of you know the CRISPR, so I'm not going to get in detail with that. But as you know, it's a very huge name. Clustered, regularly interspaced, short palindromic lipids. Oh, come on, when I first heard about it, it was too much for me either. So we know that these short palindromic repeats, they are discovered like 1987 or something like this. So they found this short palindromic repeats, but they couldn't understand what is the function of it. So it's very important. I'm always talking, when I'm talking to my students, I'm always saying them, yes, you're giving me some data, but you have to show me what does that mean. So having a data is not the important thing when you're in the lab, but when you say something about the data, it's very important. So how you become an academic person when you say something about your data and when you analyze it, doesn't matter how much you experiment that you did, you have to analyze it first. I'm always telling to my students like this. So from the CRISPR, we know that we have different Cas proteins, like CRISPR-associated proteins, but the Cas9 is the most favorable one, I guess. It's very popular, and we know most of the mechanism of the Cas9. So this is the enzyme, and we have the gRNA, we have the guide RNA. So this, as you can see, this guide RNA and the Cas9, they're working together. And they can target like 20 nucleotide base pair by using a very specific PAM sequences. It could be like NGG, and together with other Cas proteins, these sequences are changing, and each day they're finding new Cas proteins. And this short hair loop structure is highly important. Why? Because in order to activate this Cas9 and the nuclease activity, this structure is necessary. And after that, this is the spacer or protospacer place that you can go and find your target, and after that you can do some changes. So we know from the DNA repair mechanism, we have basically new repair mechanisms are coming, but we have two different options, like non-homologous end joining and homologous directed repair. So when there's a Cas9 enzyme, just the endonuclease activity, as you can see, it's work like a Caesar. And when it cuts the DNA like this in the both strand, this DNA can bind, it can come together with different combinations. And these combinations can lead to insertion of new nucleotides, deletion of new nucleotides, or the frame shift, which means that mostly these refer to non-functional proteins, because you have the frame shift, your proteins is no longer have the same code. So that's why your protein is no more functional. Or you can have a homology directed repair by in the system. Your sister, there is a cut, of course, again, And after that, this system uses the true sequence from this sister chromosome, chromatid. And by using this true sequence, it can do the homology-directed repair by recombination. So these are the methods that we know mostly, but they're very important details in those methods. If you're working in the lab, for example, you have to know we have a cell cycle, and each cells have a cell cycle. And some of the cell cycle cells mostly prefer non-homologous end joining. So if you like to do homology-directed repair, so you have to arrange your cell cycle while doing your experiment as well. But these are completely details. So not only to know the theoretic, information about CRISPR, but also, I guess not only the CRISPR, all the gene editing systems are based on molecular cloning. And molecular cloning is not very popular in Turkey, but it's very, very important if you're working with gene editing technologies. So in each field, the vector design is cloning, and cloning is very complicated. Generally, in order to do some genetic gene editing in the cells, we are using plasmids. You know what plasmid is? Plasmid is normally in the bacteria, and it's like episomal. It's not integrated in the DNA of the bacteria. And when the bacteria replicates, the plasmid replicates itself as well. So by using this very small and very working vector, we can design very different combinations in order to apply CRISPR. But while you are doing this, you really need to know what the components in your vector as well. So you have to know what are those promoters, and this promoters belongs to mammalian cell, or this promoter belong to bacterial cell, because even the promoter is highly important, because we're also in our lab, we're working with the mesenchymal stem cells as well. And we know that if you like to overexpress some genes, you can use CMV promoter, like it's coming from cytomegalovirus. But it's a very strong promoter. But if you like to, for example, we're going to talk about CAR T cell therapy, you have to work with the EF1 alpha promoter, which is not that much strong. And as a side effect, it's not inducing like the side effects of the cytotoxic T cells as well. So everything is so much in detail. People are just collecting lots of data. But for now, I guess because of the COVID, we cannot get new papers about those gene editing and gene therapies, because people are at home like us. We're always working in the lab, but it doesn't matter because most of the places are shut down. So we cannot go a little bit further. But new new therapies about this cancer, about this immune system will come eventually soon. I hope so. So during the vector design and cloning, like I told you, it's highly important. You have to know each component of your vector, and you have to analyze, or you really have to know what you're going to do with this vector. So it's easy to buy it from the Edgene. There's a website, it's called Edgene, but they're not saying you that if you're going to do this experiment, please prefer to take this plasmid. No, you're the one who are deciding to get this plasmids. And not only the plasmids, the delivery methods of the CRISPR are very important as well. And according what are you going to do, these delivery methods are depending, of course. I mean, from the basic molecular biology, we know those cationic polymers and lipid-based nanoparticles or electroporation we're using so much, but they're very toxic to cells, and their efficiency is very low. I mean, the efficiency delivery of the plasmid inside the cell. So their efficiency is very low. That's why people are evolving they're creating or developing new nanoparticles. Also in our lab, we're using some different kind of nanoparticles as well. And also this is the plasmid that you're going to clone, or you're designing. And rather than using plasmid, we know that you can buy these Cas9 protein, and you can create this gRNA by using in vitro transcription method as well. So most of the people are using this ribonucleoprotein and Cas9 complex for new gene editing mechanisms. So sorry, yes, I try to find what you're saying, but it's fine, I guess. And for the viral delivery methods, you probably heard about those viruses and adeno-associated viruses and lentiviruses. They're coming from different family, and their effectiveness is also different from one to one. These lentiviruses, they can integrate the genome of your interest in your genome. But these adeno-associated viruses, they're working like episomes, so they're not, it is said like this, they're not integrating any of those their particles inside the genome. And because of that reason, people, for the in vivo therapies, what I mean by saying in vivo therapies, you're directly injected your CRISPR materials by using these viruses through the patient. And people are using these adeno-associated viruses, but very recent results shows us that somehow we know that these adeno-associated viruses are working at the episomal condition, but somehow they integrate some of their genomic material inside the cells and lead to an insertional mutagenesis. It's just very important data, because most of the field are focused on the AAV. And so for now, people are trying to find new AAV viruses, and also people are trying to increase these nanoparticle structures as well. For example, for the COVID treatment, we know that we are having these lipid nanoparticles in order to get this COVID vaccine as well. So again, for the potential of CRISPR in cancer therapy, like I told you, there are lots of I mean, we can talk about CAR T cell therapies like whole day, or we can talk about these screens or anything, that the suppression of the epigenetic mechanisms or activating of epigenetic mechanisms as well. But they're all from very, very different fields. So I have just 20 minutes, so I'm not going to get in detail all of them. But I just want to talk about the target discovery by CRISPR screens first of all. For example, you think that some of the cancers, we know that they're having some resistance, and you're giving a drug and it's not affecting somehow. So we don't know how it goes. So which gene causing this resistance as well. So it's a very nice method to understand the resistant genes and the new targeted therapies by using these screens. So while you're doing the screens, they're like, first of all, you have to design your guide RNAs, you have to synthesize them, and after that you're making like CRISPR libraries, you're amplifying this library, and also you can buy it from like Horizon or Dharmacon as well. And after that, you're giving this library into lentiviruses. Like I told you, these lentiviruses can integrate your gene of interest into your genome. That's why in most of the studies we're working with the lentiviruses, because their efficiency is so high. For example, when we use cut unique polymers, we can only integrate like 10% of the genome of our gene of interest. We can easily see that, but when we use a lentivirus without selection, we can obtain like 90% efficiency. So it is huge, and we know that these people are not using lentiviruses too much because since it's integrated into the genome. But for now, it's not. We know that it's not making any insertional mutagenesis, which I mean it's not integrated. Some of their its part very close to the cell cycle dependent genes. So that means they're not making some mutations, or that they're not making some other, how can I say, changing the phenotype of the cell, but still it's integrated in the genome. And we know that DNA has lots of hotspots, so we still don't know how can we use these lentiviruses, but also there are like integration deficiency lentiviruses as well. People are using this because the efficiency, I mean, infection efficiency is too high. The integration is a problem. There are these kind of enzymes people are using, but their efficiency is not as much better as the antiviruses. So the field is open. You can use different enzymes, different viruses. The most important thing is efficiency, and also you shouldn't change anything in your DNA rather than your gene of interest. So after you prepare your lentiviruses, you can give it to your cells, and after that you're having this infected cells library. And while giving it to your cells, we have a term called multiplicity of infections. So what does it mean? We have viruses and we have cells, and there's a ratio between them. So the ratio between the cells and viruses. So for example, multiplicity of infection 1 is I give one virus to one cell. But in this case, in order to have different sgRNAs, as you can see in your cell, you're using very, very low multiplicity of infection levels, like MOE 0.3 or not more than 1. And because we would like to have that each cells have different sgRNA from this amplified library. And after that, you can do like positive selection and negative selections. For example, if you're just checking the essential genes, so we know that this is CRISPR and it's integrated into genome by lentiviruses. After you did this antibiotic selection, most of your essential genes will, which contain the cells containing essential genes, they will die. But for the positive selection, you can check the other drug-resistant genes by using positive selection, and always people are doing this NGS sequencing in order to determine what happened to our library system. I just want to give a very basic explanation about this. For example, people are using or taking these CRISPR screens so much, they try to find out how we can find new targets for the cancer therapy. And it's like 2015, it's very, it's not very close to our time, but they found that there is quite a little bit interaction with the BRCA genes and PARP1 inhibitor. So we know that cancers have different interactions, guys. For example, this one is I'm going to show you as a synthetic lethal interaction. That means not only one mutation is enough to kill the cancer cell, like at least 2 mutations it needs. And it's what we are having is over here like this. For example, BRCA gene, it's responsible from the homologous recombination mechanism in your cells to repair your gene. And also PARP1 is an inhibitor, PARP1 is an enzyme. It's important for the base excision repair. So they both, as you can see, working for the repair. And these cancer cells, even their cancer cells, they need this repair mechanism. We know that. So in the cancer cells, we have the BRCA mutations, but PARP1 can compensate this mutation, and it can do the repair by itself, because when you have this, as you can see, a broken DNA, it has to be repaired for the survival of the cancer cells even as well. But if you don't have any, for example, if you don't have any mutations, you're by using a PARP1 drug, PARP1 inhibitor, you just have the BRCA, it's again doing the repair. But when you have BRCA mutations, at least 2 mutations, BRCA1 and BRCA2, because if you have 1 mutation, it can compensate the other one. So it is very important for me, I guess, because at cancer, most of the people are focusing on the point mutations. Yes, there are like several therapies that working on the P53, mutations on the P53. But when you repair just 1 mutation in cancer cells, sometimes it's not enough, because it's a very complex and heterogeneous disease. As you can see, the complexity of the disease doesn't matter. If you repair the mutation, it can find another way to proliferate itself and accumulate new mutations as well. So sometimes I'm saying this, but CRISPR is not the good idea because you're knocking out the cell, knockout genes, sorry, not the cell, and you're completely make this gene dysfunctional. But when you're using like short hairpin RNAs, it's more powerful technology in order to understand what is happening in those cancer cell pathways as well. So in here, when you have your BRCA have 2 mutations, and you use the PARP1 inhibitor, they show that at the breast cancer and ovarian cancer, these cancer cells can go cell death, because we know that they are very resistant to cell death. And it's not the only one, that we have different lethality systems, like collateral lethality also. As you know, these normally these cancer cells, they're accumulating those mutations. They generally do these mutations at the tumor suppressor genes, because we know that these suppressor genes can suppress the tumor. But when you do the mutation, they no longer can suppress the tumor formation. And also it's very new, I guess it is for me, but these cancer cells, when they're dividing, they're also doing mutations that they're not related. We call them passenger genes, and they're doing mutations in their neighboring cells that they're not related with the tumor suppressor genes. And we don't know what these mutations cause. So by using this drug libraries or like CRISPR knockout libraries, people can find new drug targets for the cancer treatment. And not only this drug libraries, but also, we have lots of different functions as well. For example, transcriptional activation and repressor. For example, at the breast cancer, they know that BRCA1 promoter methylation. So in order to remove, we can remove the methylation and we can remove the genome instability, which is one of the cancer hallmarks. Or we can reactivation of, as you can see, hypermethylated tumor suppressor genes in order to evade tumor suppression, and we can remove it, this one as well. So these are the different structures. And by using this system, we are using actually the Cas9 system. We call it dead Cas9. Why it is dead Cas9? Because it doesn't have this nuclease activity at both strands. But as you can see, it can go and bring its target. It can find its target, and it can bring a very different protein. Like these are the VPR. This is an activation protein in order to activate these methylated places. Also, a TET1 is another activation protein. It can also demethylase the domains. As you can see, demethylase means removing the methylation, and it can activate the gene again. So these are highly important mechanisms, but it's not easy to design as you can see, an sgRNA, a single guide RNA for this system. They're very limited. So it's not easy to do these activation and repression mechanisms in each gene whenever you want. It's not a world like this. Also, we know from this tumor transcriptional activation, we can do transcriptional repression as well. We can repress some of the genes, we can methylate them. So we can inhibit the activity of them. And also we have epigenetic modification systems as well by using different proteins like DNA methyltransferase 3. It increases DNA methylation, CpG motifs, and repressing the gene transcription. So in order to understand what is happening when we are activating those genes or repressing those genes, it's these tools. This is what we call CRISPR-I and CRISPR-A and CRISPR-I. CRISPR activating and CRISPR interfering systems are very valuable tools for in order to understand the functions of your genes at the protein level as well. So what are the other therapies? Like I told you, we have a very new immune system therapy approach. They call it CAR T cell therapy. We know that our immune system cells, they're not able to somehow catch and find the cancer cells and destroy them. So that's why people, they're taking patients' own T cells and engineer it, like inserting a gene or sometimes knockout gene on the T cell, and they're educating. You know that normally our T cells are educated at our thymus. Before we born, our education has also, it's completely finished. But some of these T cells cannot be educated very well. So people who have autoimmune diseases when they're born, like diabetes, that means some of the cells, they're not well educated. Somehow they escape and they find a way to do this autoimmune reaction. And these cancer cells also somehow interacting with your immune system and saying to those cells that, okay, I'm your body cells, do not kill me. We don't need to do this, guy. So that's why you need to educate your T cells or NK cells. Lots of immune system cells are in this CAR T system, like Treg cells are very important, T regulatory cells. T cell, gamma delta T cell, they're already at the epithelial cell. We know that they could invade the solid tumors very well, but it's not easy to do this application. For the cartoon, I can told you like tons of things about it. But when we go to the lab, these cells inside your body, these cells are very like monster. They can do everything, but when you take them out, they're very vulnerable. When you do any little tiny application to those cells, you can lose those cells. And this is why it's not easy to work those cells at the lab. So you have to gain lots of experiences in order to do that. But saying them, it's like when I say something like this, it's like we're curing the cancer, but no, we're very at the beginning of it. So you can educate your own T cells, you can grow them, and you can give the appropriate amount to the patient. But also they have lots of side effects in the CAR T cell therapy. But people are working with this, how can I say, field in order to remove these side effects, because it's a very promising therapy, immune therapy, especially for the patients who relapse the cancer. Because sometimes people are taking drugs and they heal, but after that, when they relapse, when the cancer occurs again, these drugs are no longer effective. We know this. That's why these new therapies are very important. Or the other one is the PD-1 knockout T cells. Like I told you, these T cells and cancer cells were very perfect interaction. So this is the PD-1 is a programmed death receptor, and this is a programmed death ligand. So when they interact with each other, the T cells saying that, yes, okay, it's my cell because I recognize it, so I don't need to kill it. Oh, come on, it's a cancer cell, we need to kill it, of course. So in order to remove this interaction, we can knock out this PD-1 on your T cells, and then after that there is no interaction. Or you can use anti-PD-L1 antibodies, of course. But for the CRISPR, I'm just saying that you can use PD-1 knockout cells in order to remove the function of the this protein, the cell surface protein, and after that, your T cell can recognize your cancer cells after you give it to the patient. So like I told you, this is a huge market, of course. This is gene therapy. I'm going to talk that, but for example, this is CAR T cell therapy for just, one dose of, for one patient, it costs like $475,000 just for one dose, and one dose is not enough for a patient. So it's a huge money, and unfortunately, it's a market. So people are, and we know from the compound annual growth rate, this market is growing like 40% is too much for a market. We know that two beautiful ladies like Jennifer Doudna and Amanda Sharpenter, they took the Nobel Prize in chemistry in 2020. It's a very, very huge, because that's very early, right? Normally people, when they're getting very, very old, taking this Nobel Prize, but they saw that this CRISPR can be applicable not only at the gene editing, but on the industrial systems as well. So, and many systems. That's why, because it's of this coverage, they gave the, and also it's easy when we talk about, I didn't talk about the other nucleases, programmable nucleases, but it's very easy to be applicable and design. You can even design your own plasmid in your lab, and you can do your experiments, and you know it. So it's like a 7 billion market. So we have.
Sorry, I interrupted you, but we have 5 minutes left. 5 minutes.
Okay. And we know from this CRISPR, these Emmanuel Charpenter and this Jennifer Doudna have their co-founder of their own companies. And these CRISPR materials, like we are taking plasmids and everything from ABN Group, Thermo, and Horizon. We know that Turkey doesn't have any participation to this market since that time, which it encouraged us to startup for our startup. Now we have a virus and biotechnology startup, and we are just 3 people, like Özgür Tataroğlu and Erdal Karoz. We're just in the system. So it's a very new startup, but in the startup we can offer lots of like gene editing services, like the vector plasmid viruses, high custom lentiviruses, lentiviral kits, luciferase test kit, GFP test kit, and also we have other products and services. I mean, it's huge for a 1-year biotechnology firm, but we have still upcoming products like Cas9 protein, RMP, and mutant tech protein, or MMLV. So we can create knockout cell lines. We have like huge data I can show you, but just for like give you an how can I say inspiration. We're always working with those cancer cells like human embryonic kidney cells, ACT cells, colon cancer cells, lung cancer cells, whatever it comes to your mind, and we can easily knock out the cells, or we can do the knock-in or overexpression of the genes by using our products at the molecular level, and we can check the functions of the genes. We have like academic projects as well, but for now I just talk about our company because I don't have lots of data to show you. I'm very at the beginning of my, how can I say, career. That's why we're collecting data about our projects. So if you have any questions, you can always reach me from our website or from the LinkedIn, and also you can send me an email. Thank you for listening to me, and I can get questions.
Thank you, Doctor, for this informative speech. If there is any question, you can write on the chat part and we can ask the doctor. No question?
It seems there's no question.
Okay, thank you so much, and then have a nice day.
Thank you hocam.
Thank you.
We should take a break for 10 minutes. Please don't start the meeting. Just we will close our cameras and microphones. Please be here. We will continue after 10 minutes. Let's say 2:15. Is it okay for everyone?
Yes.
Okay, let's give a break. Then we will continue with the second session.
Şeyma'cığım sesini aç.
Welcome again. We will continue with the second session, and we can move on. The new topic is the plant pathology with Associate Professor and Dr. Emre Yürük. He will present it. Dr. Emre Yürük completed his master degree and doctorate at Istanbul University in molecular biology and genetic field. He has many scientific papers and he has a rich research background. And he is currently studying at Istanbul University as associate professor. And we can listen to Emre Yürük. Please, Dr.
Yes, thanks for inviting and also introducing me. Firstly, I've tried to share my screen. Is it okay to hear my voice also?
We can see your and we can.
Yes, if you can speak now. Okay, my speech is on about plant pathology and in particular to fight against the phytopathogens belonging to fungi. At a historical aspect, in the year 1845, there was a great famine in Ireland. It was caused by several names: Gortamor, Büyük Açlık, Büyük Kıtlık, Great Hunger, Famine, or Irish Potato Disease or Irish Potato Famine. It led to approximately 30% of the country's population decrease and also emigration to America. Also, this famine permanently changes several topics of the island's demographic, political, and cultural landscapes caused by just a fungi phytophthora infestans of the potato. In phytopathology, in particular, struggling with the disease and the fungi, plants are exposed to several biotic and abiotic stress factors, including humidity, disease, viruses, and bacteria, and some others. Plant pathology is related to disease developed by and attack of some microorganisms with the specific conditions. Morphology, physiology, diagnosis, epidemic resistance, and management are the important topics of the phytopathology investigations, and not only attractive and aggressive phytopathogen is needed, also environmental conditions and the susceptible hosts are also needed for the host becoming the safest. In phytopathogens, you can see several literatures about divided into several groups, microorganisms such as 4 main groups: viruses, bacteria, fungi, and others, or 11 distinct groups. We can see, but the major causal agents are belonging to Ascomycota with the fungi with hyphal development, and natural habitat is the soil. Yeah, there are several times we see on TV channels some news and reports. Cancer for plants. Yes, there are, have you ever heard about? I don't know, but maybe you have heard it. Yeah, there are cancer for plants. Yeah, but what? I'm introducing to natural genetical engineer, Agrobacterium tumefaciens and Agrobacterium rhizogenes, which are causing the disease called as chrome gall, dicotyledon especially, and the crazy roots of Agrobacterium rhizogenes. Okay, this is like the tumor-like process. And Bean et al. Report that there were top 10 fungal phytopathogens, including Fusarium graminearum, Ustulagomyces, and some others. But this is the variable according to the authors. But maybe this is the pre-knowledge for us. Life cycles are so and so interesting, but we can easily say that there are 3 different life cycles of fungi for, in particular, phytopathogens, necrotrophs, biotrophs, and the combination of these 2 strategies, hemibiotrophic microorganisms, which was the most harmful for the plants. We can see some differences given in the list, systemic being differences or narrow host range or wide host range, such as, for example, belonging to the same genus Fusarium graminarum and Fusarium culmorum. Fusarium culmorum, culma is the meaning that stalk. With this necrotrophic microorganism could attack any plants. But graminarum, gramina is meaning that the serious, such as these plants, and narrow host range. We can easily distinguish these microorganisms according to their life cycles. And in particular to management of fungi disease, which strategies can we use? We can 5 main strategies. Here we see fungicide treatment, most powerful strategy. We can easily say that, and the crop rotations, the traditional techniques, and the disease-resistant cultivar usage. It could be divided into 2 fields as naturally occurring disease-resistant cultivars and also genetically modified organisms. We can say the novel agents usage or treatment. We can say that polyaromatic hydrocarbons or plant-derived essential oil treatment is also some important examples of novel agents, and biocontrol agents such as Trichoderma sp. What about how can we plant disease management, doing plant disease management, avoid them from pathogen, excluding it, and if it's possible, destroy it without not distracting the ecological area. If it's not possible, we have to save the host, and we have to develop the resistance, and if it's not possible, therapy, the last step. Stubble burning, Anus yakma. Here we see left top side, and one of the most common fungicides, thiophanate methyl, we can see, and the VIGS strategy for developing the genetically modified plants in use. Struggling with virus and bacteria, what about? First, we can use meristem cultures, not including virus, but this is really not so easy. Taking the meristematic shoot segments of the plant's apical region is so laborious technique, but it's possible. Removing this plant from the fields, yes, it's so clear approach, and blocking the vectors containing viruses, yes, it's also okay, but it will be late after disease development. And the genetic engineering methods. What about how a pathogen could get a successful attack? First, they have to recognize the host system and should produce several factors, including enzymes. It should await from the recognition system, and it must use the plant's nutriments, such as graminarum or magna portorhiza, which could be accepted as some biotrophic or hemibiotropic microorganisms. Strategies for development of transgenic tolerant plants, transfer of several genes, or with producing proteins with the toxigenic effects of pathogen. Defense protein overexpression, transferring the genes related to phytoalexin synthesis, and expression the proteins over killing the factors produced by pathogen. Diagnosis first, we have to, after the diagnosis of diseased plants, we have to firstly identify the microorganisms, the causal agent of the diseases. Fungi are generally divided into 4 groups, and additionally 3 groups were also reported from the literature, but there are also dilemma in diagnosis of fungi specific identification. Example given here, please see more than just as a sample, 200 toxigenic Fusarium strains have been wrongly identified in literature. And in identification of pathogenic organisms, which strategies we can choose or we can combine. Sequence characters, amplified region markers, yeah, is one of the most common strategies. And multilocus genotyping is not cheap, not expensive, but we can combine it by add in the morphological tests or NGS technologies. Genealogical concordance assays accompanied with flow cytometry. Yeah, it's also used in, and the chemotype and serotype assays were also used in diagnosis. As a sample here we see the diseased kernels of barley, and we take the diseased kernels from the spike, and we allow fungi to be grown for 7 days, and then at microscopic level we see the microconidium, and we get monosporic cultures. Then we take DNA from the fungi, and we have to ask some questions such as is this real fungi or bacteria or another, or is there any contamination? We can amplify the 28S rRNA region, and then we have to identify the genus. Yeah, is this Aspergillus, Magnaporti, or another one? Yeah, here we also do specific locus or scar markers, and then we have to merely identify the specific organisms. Is this Fusarium graminarum or Magnaporti or another one? And we have to confirm it by we have to confirm PCR results by sequencing, by or genealogical concordance assays, or in other words. Coming back to this resistant plant cultivar development, as I said before, there are two strategies. Naturally occurring is the first one, and the genetic engineering strategies is the second one. But these are also ecological and economically important strategies. But the limited agronomic traits is one of the most important disadvantages of this technology, and long F3 population is needed. That means at least 3 or 4 years. This was also disadvantages of the disease-resistant plant development. Entrapment of antagonist microorganisms. Yeah, this is ecological approach, not expensive, non-laborious, but there are limited knowledge about the mechanisms between the two microorganisms, these friendly microorganisms and phytopathogen microorganisms. We have limited knowledge about the epigenetic or oxidative stress or apoptosis related mechanisms lie behind, is not clear. And some of important Cryptococcus, Trichoderma, Pseudomonas, and Bacillus sp are some of important antagonist microorganisms. Here we see left-sided, sorry. Here we see left-sided petri dishes on control cells, and the right-sided are also decreased radial growth rate of the phytopathogenic microorganisms. And the most we like it, anyone like is fungicide treatment and fungicide usage are the most frequently used strategy in fight against fungi, and some others. Cost-effective, non-laborious, easy to application. There are several advantages of this strategy. Here we see some examples. For example, thiophanate methyl targets the beta-tubulin protein and degrades the skeleton, but no renewal of the strategies or using the same combinations of the same fungicides are let fungi to be more stronger than before. For example, here we see the 198 position for mutation in beta-tubulin sequence of the Cercospora beticola. And then we see that the fight fire against the fire or ash to ash, dust to dust by rise by fungi. Yes, here we see some other fungicide targets such as respiration, signal transduction, sterol biosynthesis, ergosterol 11. For example, cell wall biosynthesis, unknown mode of action, nucleic acid metabolisms, and some others we see. And another promising strategy was plant-derived essential oil compounds or polyaromatic hydrocarbons. These two are promising ones, but as I said, for antagonistic microorganism usage, there are unknown mode of actions still. In last 2 decades, the increase of the papers published, we easily see that the use of specific compounds such as camphor, terpinol, and some others here we given. But the disadvantages of this approach is the usage of high amounts of plant-derived essential oil compounds in comparison to fungicides. As a general, we have to first identify the disease plants, and then we have to transfer them to laboratories, and we have to identify the causal agent at species level, and then we have to get more information about it. But in field and in implanted treatments, we in general use the fungicides, but it's apparently clear that we need more ecologically and more friendly strategies, such as plant-derived essential oil treatment in fight against plant disease. Here you will see the references that I used here. Also, you can download the PDF file for 2 alternatives, and I would like to thank you for this organization, and you can always send email from this email address, embryo@outlook.com.
Thank you, Doctor. Is there any question? Doctor, by the way, I want to ask a question. May I ask? I saw in the presentation quercetin.
Yes.
Is it related with your topic, or I'm mistaken?
Yes. Yes, sure. About 3 years ago, I started to investigate which plant-derived essential oils could be effective on Fusarium graminarum or another Fusarium species phytopathogenic fungus, and I see that quercetin is not investigated by this term. Yes.
You mean you are extracting essential oil from the plant and you are applying it in a diseases, right?
Yeah.
Am I wrong?
My collaborators are extracting, but I generally do genetics tests. But additionally, we sometimes need specific compounds such as quercetin, alpha-2 and others from commercially. We can buy them. We also buy them.
Okay, thank you, Doctor.
I thank you very much.
Dear participants, I think there is no question, right? Yes, it was really informative. Thank you. I think we can go on to next topic with Dr. Zeynep Büşra Bolat. Büşra Hocam.
Yes, hello.
Okay. Here.
Can you see me?
Yes, Hocam. We can see you. Welcome.
Thank you.
The new topic is the targeted drug delivery in cancer therapies. The doctor is Dr. Büşra Zeynep Bolat. She completed her master's degree at US Technical University and completed her doctorate program in Yeditepe University. Her field of interests are molecular cancer biology, targeted drug delivery, and the drug, the presentation name is targeted drug delivery in cancer therapy. So we can continue with Dr. Bolat. We are listening to you, please.
Okay, thank you, Şeyma, for the invitation also. Let me share my screen first. Okay. So hello, everyone. Again, my name is Zeynep Büşra Bolat. As Şeyma said, I'm from Sabahattin Zaim University. Can you see my screen?
Yes, Doctor, we can see and we can hear you as well.
Okay, good. So today I'm going to talk about the targeted drug delivery in cancer therapy. This was mostly the study was done during my PhD and after the postdoctoral when I was doing in Yeditepe University. In Sabahattin Zaim University, I started in Sabahattin Zaim University at 2020 in April, so we have a lab there, but mostly the study was done with during my postdoc study. So what is cancer? As Professor I just said, what is she went in details of what is cancer and how it's the hallmarks of cancer. I'm not going to go in details. Cancer is the abnormal growth of the and division of cells and is still a major public health problem worldwide. And in 2020, there was still estimated as 19.3 million new cancer cases with almost 10 million cancer deaths. So the early prognosis of the diseases due to the screening and better understanding of the pathophysiology of the tumor progression has been open new vistas as therapy options. I'm not going to go to details of the hallmark of cancer. So what are these options, the therapeutic options? Now in solid tumors, after the surgical removal, the remaining cancer cells are done either radio, chemo, or immunotherapy. However, once the cancer metastasizes, the treatment options are limited and the chemotherapy remains one of the choices of treatment. And the reasons, one of the main reasons for failure of the chemotherapy is poor accessibility of the antiaplastic agents. In this case, it could be the chemo drugs or the requirement of high doses. So you need to give high doses and non-selective nature of these agents, which causes severe toxicity with the remaining body cells and tissues. So in cancer treatment, where chemotherapy or radiotherapy options are designed to kill cells, the specificity of the drug action gains still important. So the idea, the strategy that behind is to preferentially to kill the cancer cells without giving any toxic effects or any side effects and giving any significant toxicity on the normal, the healthy cells. It is necessary that all cancer cells must be killed either the direct way or indirect way. So in order to achieve the complete remission in patients presenting the disease. So here I'm going to talk about the drug delivery systems. The standard chemotherapy administration leads to drug resistance and can become inefficient of time. In case of doxorubicin, which is used back in, I think it was 1980s or more than that. So while if you're administering doxorubicin alone, it showed that cardiotoxicity to patients, killing patients afterwards as a side effect from cardiomyocytes. So the drug delivery systems encapsulating the drug, the chemotherapeutic drug into the system extends the viability of the drug, the administration drug is reduced, it prevents the drug side effects. And when you look at the features of the drug delivery systems, as we said, should be non-toxic, monomeric, and additives. It shouldn't have drug stabilization. It should have drug stabilization and solubilization, protecting the drug, and good biocompatibility, which is also important. It should have a prolonged circulation time. The drug should be on the system in a long time. Controlled drug release, which is also very important. It should be tissue cell specific targeting, penetrating of biological barriers, biodegradability and biocompatibility is very important, and shouldn't give any cytotoxic or immunogenicity. And for excretion by livers and kidneys, no accumulation in the organs. So you don't want the drug itself to give toxic effect with the remaining of the body, where people who have extensive chemotherapy have kidney failure problems and other problems. And we look at the clinic point of view, it should be safe and easy applicable. It should have a good cost, risk, and use profile, easy to store and sterilize. So these are all the issues, all the features you have to overcome to have a safe, a good drug. Now, when we look at the drug delivery systems, this could be in liposomes, different nanoparticles that could be given. One of them is liposomes, which are small sized nanoparticles, which have phospholipid composition. This phospholipid composition is important as it gives, it shows biocompatibility, biodegradability, low toxicity, and low immunogenicity characters. And it is known for the enhanced accumulation in tumors and long systemic circulations. And liposomes are used as nanoparticles to encapsulate the drugs. They have advantages as it increases the solubility, prevention from premature release into bloodstream, and protection from degradation. For polymersomes, which are also another nanoparticles, which is aqueous amphiphilic copolymers assembled into polymersome structures. Here in the picture, you can see a polymersome structure, which they have loaded a hydrophilic drug and hydrophobic drugs. So it gives a good option, as you can load either if your drug is hydrophilic or hydrophobic, you can load it. And here two homing devices. So these are the, in this case, they showed as antibodies, but these are the targeted ligands that binds to the receptors of the cancer cells. So polymersomes have the advantage of being more flexible than micelle structures and have ability to deform to pass through narrow passages. Polymeric polymersomes made of either natural or synthetic polymers have high stability, easy changeable polymer characteristics, and surface chemistry. So these characteristics give them great advantage to be used in controlled drug release. As we look at the micelles, these polymer micelles are amphiphilic block copolymers, which form pore shells, one left in aqueous solution. So in this case, these are micelles, these are liposomes. So you can see the difference between a micelle and liposome structure, carrier. And micelles have advantages as increasing permeability and the half-life of drugs, which is an also important feature for drug delivery systems. Micelles fabricated by antibodies are used as the HER2 positive breast cancer. Taxol drug delivered by the HER2 antibody micelle complex showed high efficacy in comparison to lipid-based proteins. So loading the drug into micelle structures and targeting with the antibody gives a good efficacy in cancer therapy. So here you can, of course, these liposomes, micelles, and polymersomes are not only nanostructures. There are carbon tubes, iron oxide nanoparticles, dendrimerase micelles, gold particles, and silicon nanoparticles, and quantum dots, which are used. And there are a lot of studies with these nanoparticles too. But this study that I'm currently talking about was a 1003 TÜBİTAK study. So we mainly focused on different carriers with liposomes, polymersomes, and micelles nanostructures. So for loading drug, with the drug we used was doxorubicin, which is an FDA-approved drug used a lot widely in cancers like ovary, lung, breast, stomach, and it acts in cancer cells by either intercalating into the DNA and causing disruption in topoisomerase 2 or generating free radicals which damage the DNA. studies show that when the doxorubicin was administered freely to patients, as I said before, they showed cardiotoxicity. However, liposomal doxorubicin had less cardiotoxicity to patients compared with administration alone. So this gives a great advantage. And there are so many doxorubicin formulated with liposomes. Here there's Celts, Doxil, and Myconet, which are all approved in phase studies. And new studies shows that phase 1 study as the anti-EGFR immunoliposomes. So this is a targeted study, which is also in liposomes and useful in solid tumors. So what are these smart targeting? Let's look at them. Now, for targeting, there's also passive targeting and active targeting. Okay, so when we look at the mechanism of passive tumor targeting by nanoparticles, actually, there's another one as physical targeting, which I didn't put here. So physical targeting goes with magnetic field or ultrasound particles, but we'll focus here in the study as the active targeting, which was important. So passive targeting is when the drug is delivered by EPR effect, reaches the tumor site. However, in active targeting, there is ligands that are bound to the nanocarriers, so they go directly to the tumor, to the specific receptor that is in hand. In active targeting, there's antibodies, small molecules, aptamers, and peptides used. In our study, we use peptides as the tumor homing targeting. So when we also look at clearly the receptor nanoparticle, which is targeted with the peptide itself, goes and binds to the receptor, and by endocytosis goes into the cell. So as most of maybe you know, trastuzumab is one of the main drugs, the target antibody that is used. Now, the pioneer research in using ligands for target drug delivery began with antibodies, but there are some limitations, which is the antibodies are large in size. There is a possible immunogenicity, low physiochemical stability, and short in vivo half-life. So tumor targeting peptides gives advantage as they're smaller in size. They have excellent tissue penetration properties. They can be easily chemically conjugated with drugs compared to the antibodies. And these tumor home peptides are polypeptide-based targeting drugs. And the tumor homing and tumor target peptides, these are all similar technologies, are some of the strategies used for targeted cancer cells for delivery of chemotherapy drugs, as well as diagnosis of cancer. So here also it is used in diagnosis of cancer, not just loading the drug and delivering it to cancer. Some of the peptides that are used are PEP1, which is for pancreatic islet capital endothelial cells. These are used, this study was done with PGL, PEG nanoparticles, and for RGD, which is used in targeting the alpha beta integrins. There are different nanoparticles that are used. So one study was done with peptide P53, which targeted the prostate-specific membrane antigen in prostate cancer. Okay? So what we did was that, so another study done was shown that P53 was a good peptide targeting the prostate cancer, and then LNCAP cells had more expression compared to other cancer cell lines. What we did was that we checked whether we could see if polymorphosomes nanostructures could be targeted with P53 and could be used as in delivering different agents. So here you can see the HPLC chromatograms showing that the TERMA labeled, which is gives an orange red color with peptide and polymerisome structures. And here in the confocal microscopy, the results show that there was a strong binding affinity, a strong binding in LNK cells, which showed very less in PNT1 cells, and flow cytometry results also confirmed that there was a high binding affinity in LNK cells, showing specificity of these targeted targeted polymerisomes. And for another study, what we did was we checked for breast cancer cell lines. There were peptide 11 and peptide 18, which were used in MCF7 and triple negative cell lines. And sorry, the other article. And another article showing that MC10 peptide, which was used in which was used as a target to HER2 overexpressing cancer cell lines. So what we did in our study was that we took MC10, peptide 11, and peptide 18 together and see the effectiveness of these, compare the effectiveness of these peptides. And we have known that peptide 18 binds to keratin1 of keratin1 cells. So the study showed that keratin on breast cancer was overexpressed more than healthy cell lines. So in our study, what we did was that these are the flow cytometry results showing two different cancers, breast cancer cell lines, 565 and SQBR3, and two different healthy cell lines, which is MCF12A and MCF10A. And we used also HUVEC as also control cell line. And from here, we have checked the three different peptides and seen which one was most effective one. And we have seen that peptide 18 showed higher binding when compared to the healthy cell lines. And confirming our flow cytometry results, the confocal microscopy with labeling the peptide with TAMRA shows high binding, strong binding in the peptide 18 and 11. So what we did here also, we checked that if these can we target breast cancer with polymerisomes, and can we encapsulate doxorubicin also with effective targeting. And here Umut from Ankara University, TÜBİTAK Takman first designed the peptides, and they have produced the copolymers and given to the Ankara University, which had done Umut Janus had assembled it into polymorphism structures. So these red signs here are the peptides itself, okay, and the remaining are the parts of the polymorphism structures. So the targeted peptide 18 comes to the ends of the polymorphisms. First, we had to see if this polymorphism structure was cytotoxic to cells, so MCF10A, and for HEC, VEC, and mesenchymal stem cells, we have seen that there was only slight toxicity in HEC 2030 at high concentrations and at 72-hour time point. Then we have, Umut has checked the cumulative DOX release, seeing that whether comparing the free DOX and the targeted polymerisome encapsulated in, doxorubicin encapsulated in targeted polymerisomes, the release profile showing that there was a prolonged release in encapsulated targeted polymerisomes, which was in 24 hours. However, at 4 hours, the cumulative release showed that the free DOX was done. Next, we had checked that whether these targeted polymorphism, yes, we had seen that peptides were targeted, but are the polymorphism itself also with targeted? The targeted polymorphisms also show specificity. Yes, it did show specificity to AU 565 cell lines. And then we have checked whether the targeted and non-targeted and targeted and free docs showed the cellular uptake of it in flow cytometry. So here we have seen that in targeted polymorphisms, there was 81.7% release at 2 hours, which didn't show in which showed 7.6, cellular uptake in MCF10 cell lines. So we can say that yes, our structure can be used as targeted drug delivery systems. Also, we want to see whether different drug delivery systems also show good results. So we have looked at liposomes. Takmam group, Sevgi Gürüz, had conjugated the peptides with the polymers, and here we can see that these green line schematic view of the green are the peptides, and these are the dope is the part of the petoxan dope is the liposome formulation. And we have checked that these didn't also show cytotoxicity of NCF10As, these copolymers. And then Burcu Dervin from also Ankara University in the liposome group had encapsulated the doxorubicin and formulated the liposome itself. So here first we checked whether these copolymers were also effective in HUVEC, mesenchymal, and HEX cell lines. We had seen a 72 light significant toxicity at high concentrations. So the concentration of the usage of the liposome was important. And for the targeted therapy, we had seen the complex and flow cytometry results. You can see that the targeted liposomes were targeted to AU565 cancer cell lines, giving only slight binding in the healthy cell lines. Then the cumulative release of the liposome with doxorubicin loaded was checked. Also here, similar to the polymorphism study, we had checked if the liposome itself, the liposome without targeting and free doxorubicin was effective at being a targeted drug delivery systems. Yes, again, we had seen that there was a high cellular uptake in cancer cell lines compared to the healthy cell lines. However, in free dogs, there is a lot of cellular uptake of the drug itself. So also here you can see a schematic view of our liposomes with peptide 18. So and the loading of the drugs itself. We had also seen the cell viability results showing that 2.5 microgram per ml of the dogs showed non-significant at MCF10A. However, with high dosage, the targeted therapeutic effect was less. So here you can see that these are the cancer cell lines, these are the non-cancer cell lines. At 2.5 and 1, there isn't a significant effect if MCF10A compared to the control cell line. However, with the high dosage, there is a lot. So the dosage we had to use was very important. What we did next was we did
5 minutes left, I think. 5 minutes left. The last 5 minutes.
Okay, thank you, Shema. So I'm going to wrap up. So next studies were the in vivo studies, which are important. However, these studies are still under review, so I couldn't put them in this presentation. And when we look at the cellular uptake of the doxorubicin-loaded doxorubicin-loaded liposomes, targeted liposomes, there was still at 48 hours, there was still clear cellular uptake of the doxorubicin. And for the in vivo results, we had seen that there's a significant difference compared to the control mice. So I want to thank TÜBİTAK 1003 because this project was mainly supported by them, and Yeditepe. We had done this under the premises of Yeditepe University, and my supervisor Fikrettin Şahin and Dilek Telci and Ece Nezir. This is one of the photos from the in vivo studies, which we had done for hours. And the polymer group, TÜBİTAK MAM, Özgür Yılmaz, Sevgi Gülyüz, Umut Uğur Özkese, and the polymerzome group, Asu Bozkır, Umut Canöz. And from the liposome group with Burcu Devrim and Ongun Saka. As I said, most of the in vivo papers are not still out, so I couldn't put them, but maybe hopefully within this year, they will be out. This is our lab in Sabahattin Zaim University, Molecular Cell Biology Research Lab, which we are also doing different studies. And thank you for listening.
Thank you, Doctor. Is there any question? You can write on the chat board. Doctor, I have a question. Okay.
Okay.
Actually, not about targeted drug delivery, but about the cancer therapies. It's an idea. Is it possible to, if we add another agent, if we combine with the drug, like antioxidant, can we make the drugs more effective? Is it possible?
So you're saying that you want an antioxidant agent, you want to load it in the nano drug?
Yeah.
And you want to see if it shows the synergistic effect, for example, right? Exactly. With the drug itself. I'm not sure for antioxidant, but there are some studies done showing synergistic effects like curcumin, which is a phytochemical, which is good for your curcumin shows anti-cancer effect, right? There is also rosmarinic acid and different phytochemicals. There are so many studies that co-deliver these like paclitaxel or doxorubicin and different chemotherapies together showing synergic effects. So it's correct, yes, there are studies, and I'm not sure about antioxidants. I didn't see which antioxidant you're talking about, but I have seen a lot of studies with phytochemicals, and co-delivery studies are so popular now, which shows good results too, and synergic effect of the drug.
Thank you.
Yes, I think it will be possible, I think.
Thank you. It was very informative. Yes, we can move on with another professor. Hocam, if you stop the share screen,
okay,
we'll continue.
Okay, pause share, I think, right?
Yes.
Okay, thank you, Şeyma.
You're welcome, Hocam.
Thank you, Büşra Hocam.
Still, you're sharing your screen, I think.
Okay, I'm sorry. How should I stop? I paused.
Let me try. Stop video not. Actually, maybe at the top of the screen you can show stop screen, or at the downside. Like
I think
yes.
now, right? Okay, I'm so sorry.
Okay, thank you. And the new is Majid Haji Hussein Ali from Iran. Let's give brief information about him. He completed his master's degree and doctorate at Sharif University of Technology, with a rich academic background in biomedical, mechanical, and industrial engineering. He is the founder of and CEO of Omid Afarinan, the first bioprinting company in Iran. So we will listen a presentation about 3D printing, bioprinting. We are listening, Doctor. Yes, please.
Thank you very much. Thanks for giving this opportunity to have a talk about bioprinting and what we do in Omid Afarinan in bioprinting technology. Can you see my screen?
Yes, we can see and we can hear as well.
Thank you very much. My name is Majid Haji Hussein Ali. As you said, I received my degree from Sharif University in Iran. I had some experience in, some startups, but during last years, I focused on bioprinting technology. Let me turn off my video for bandwidth problems.
Doctor, your screen has gone.
What I said in this presentation is the first is about bioprinting, then bioprinting technologies. I will talk about bioprinting applications a little with some examples, and finally I will say about our activities at Omid Afarinan stuff. One of scientists' dreams is to create a human tissue outside human body. I mean in lab. Scientists like to create tissues in lab for implantation in the patients who need organ or tissue transplantation. From about 50 years ago, when tissue engineering introduced, some methods have developed for creating tissues in lab. That you can see some of them in the right of the picture. For example, electrospinning, freeze drying, casting, and so on. Through these methods, a process scaffold created and then cells seed on this scaffold. And you cannot control the geometry of porosities, and you cannot use more than one cell types in these methods. On the other hand, we know that human tissues have very complex geometry. Human tissues contains multi-cells and very complicated. 3D bioprinting comes to solve these problems. This video shows Dr. Anthony Atala from Harvard University that shows the capacities of bioprinting during a TED Talk in 2009. He showed 3D bioprinting technology by printing a model of kidney using a forehead by printer. Bioprintings provide a lot of advantage compared to other tissue engineering approaches. For example, you have the precision in positioning cell types, and you have potential to engineer complex tissues. And for example, you can vascularize, you can solve vascularization problem, and you can scale up easily for clinical issues. Like other additive manufacturing methods, 3D bioprinting is a layer-by-layer procedure. Tissue models create layer by layer using biomaterials. We called biomaterials here as bioinks. Bioinks may contain cells or may not contain cells. And different bioinks print next to each other to create a complex structure of different cells and biomaterials that mimicking complex structures of human tissues. Therefore, we have three main components in bioprinting that they all work together: cells, bioinks, and the device that do the printing that is by printer. According to a Henrich study, bioprinting technologies can be divided into 5 categories: stereolithography by printing, inkjet by printing, laser-assisted by printing, extrusion by printing, and some hybrid methods that combine by printing to other tissue engineering methods. For example, electro-spinning by printing. In SLA, and also it's like very similar to DLP by printing, there is a vat of resin. Here I mean a biodegradable polymer that may contain cells. The bed move in the vat in Z direction, and the source of light that is UV light in SLA and visible light in DLP expose on the surface of the vat and cure the pattern, as you can see in the video. And the curing made the resin solidified, and the build surface moved to vat layer by layer to create a 3D model. In each method called droplet, in some companies, there is a micro valve opens and closes very fast. The pressure is applied on the bio-ink tank. We call it cartridge. And by opening the valves by ink through to the bed of print, and this method is ideal for low viscosity and cell-laden bindings. In laser-assisted method, there is a beam of laser exposure that crosses through a lens, and it absorbed with a layer of bio-ink, as you can see in the video, and the bio-ink through to the platform of print. This method is also ideal for low viscosity biomaterials, especially for thin layer sheets. For example, a skin, or by printing a skin is ideal. But this technology is very complicated, and less companies commercialize this technology. On the other hand, extrusion method is the most common method of printing that completely commercialized. In this method, bio-inks print using the pressure applied to the tank of bio-inks. The pressure can be applied using pneumatic actuators, or a screw, or a piston, and the bio-ink print on the surface. Finally, there are some other hybrid methods, as I said, that combine different tissue engineering approaches. For example, in electrospinning by printing, called also electrowiring, nanofibers travels from the head of cartridge to printing platform, and they use electrical field that its voltage maybe it rise to 20 kilowatts. In the following, I want to talk about some applications of bioprinting. Because of time limitation, I will present only one example for each applications, but you may find hundreds of examples and articles by searching the net. I want to talk about 4 main applications of bioprinting. The first one is tissue engineering. I mean printing some tissue or organ for transplantation in human body to replace a damaged tissue. The second is drug discovery. I mean printing tissue models for evaluating drugs. The next one is personalized pharma. That means printing tablets or capsules with specific components or release rate. And finally, using bioprinting for printing actuator like human muscles that can be used in robotics and electronics. The first application I want to talk about it is drug discovery. In vitro tissue assay faces lots of shortcomings. For example, we need animal to sacrifice or donors to have a tissue for test. it's very complicated, and its reproducibility is very low. You may ask that we can use two-dimensional cultures or spheroids for drug discovery. Because these models cannot provide a complex geometry or control the position of cells that we can see in human real tissues, these methods cannot simulate tissue functions correctly. Moreover, for applications like skin production and test, there is some regulations from Europe that forbidden using animals for testing drugs and cosmetic materials. For these reasons, alternative models must be evaluated, maybe using bioprinting. But how bioprinting can help us in this field? Bioprinting is an ethical, animal-free approach. Bioprinting can produce tissues that biomimetic in human environment very well. It increases reproducibility. It can be scaled very easily. Bioprinting can print equivalent tissues in multiple based platforms that used in drug discovery very well, and there is no concern for tissue availability and heterogeneity. For example, here I want to mention an example from bioprinting in drug screening. Researchers from Zurich University developed a platform for the engineering of muscle and tendon tissue. First of all, as you can see in the picture, they developed a special multiple plate that contains a stand from a polystyrene. This polystyrene stand put in the multiple, and then in the next step, the tissue print on this stand. As the stand can be seen in figure D. This stand also used for keeping the model after printing suspended in agarose media. They use two bioinks for bioprinting the tendon and muscle tissue model. First bioink is the combination of pectema and gelma. They are photocuring bioinks. And the second bioink is a combination of cell-laden bioink for human fibroblasts and tenocytes. The model printed in the shape as you can see in figure A. The shape is the same size as the muscle of mouse that's used for in vitro tests in drug discovery. The model consists of a 5-layer decellulose bioink, the first bioink, and cellulose, and 4 layer of cellulose bioink. Cells were jet to multiples, and then MTT assays and some other assays done on this model. As you can see here, figure F and figure G shows collagen type 1 staining 7 days and 9 days after printing. As you can see, we can see collagen type 1 very well. Figure E also shows a stained muscle fiber in muscle section of the model that completely these myofibers completely formed in 7 days after printing. This study shows that how we can use bioprinting for automated fabrication of similar tissue models for drug screening. This shows that bioprinting is completely repeatable and can be used for drug screening or disease investigations. The next application is application of bioprinting in tissue engineering. As you know, autografts and allografts are still considered as the goal, treatment in clinic for replacement of damaged tissues and organs. There are some concerns about autografts and allografts. For example, they are not available always. They are not always available, or there are some immune response they may some immune response in patient body, and they are not suitable for critical defects. I mean the defects that are very large. Tissue engineering provides an alternative approach for autografts and allografts by the combination of cells and 3D biomaterials. But what features we expect from proper engineered tissue? What are the requirements? We need different materials and cell types and control properties in a special organization. That are the most important features for a proper tissue model. 3D bioprinting can help us in this field. 3D bioprinting allowed control geometry through computer software and therefore can be print patient-specific structures. Bioprinting allows control deposition of multi-cells and multi-materials, biomaterials, and living cells. Therefore, we can match anatomical requirements as well as biochemical and cellular components in tissue engineered, tissues and organs. For example, in a study published in Advanced Science, researchers developed an engineered patient-specific heart. It's important that we know that it's only a proof of concept. This study shows only a proof of concept for printing heart. First of all, a biopsy of omentum membrane of the patient used for extraction of stem cells and then differentiation to cardiomyocytes and endothelial cells have done. Then extracellular matrix is processed into a personalized hydrogel. And then two cells that are separately combined with this hydrogel to form two bioinks for printing some part of heart. To personalize the geometry, they use CT image, but CT image cannot provide a small blood vessel. Therefore, to ensure adequate exposure of the entire cardiac tissue to oxygen, smaller blood vessels were added to the basic vascular design, as you can see in the right picture, using mathematical modeling. Then personalized hydrogel derived from omentum mixed with cardiomyocytes and also endothelial cells mixed with gelatin, because gelatin provides as a sacrificial bioink, and this bioink will wash out to create vessels. With incubation at 37 Celsius, the blood vessel formed and the cells adhere the edge of the basic hydrogel, while gelatin become liquid and wash out. In these pictures, you see the results. In top picture, you can see fabrication of vessel shown that with cardiomyocytes and endothelial staining. Below picture show the transplantation of this cardiac tissue to omentum of a rat. It shows very well integration with rat tissue. Here is the a video from 3D bioprinting of this patient-specific heart model. This is a video of proof of concept, and the semi-transparent gel that this print have done is a support gel that is from gelatin microparticles. 3D printed pharmaceutics and tablets are the next application of 3D bioprinting. Conventional tablet manufacturing have some limitations. For example, you have the same tablet, drug dosage, and distribution of drugs in a tablet for all the patients. But here, bioprinting can help us and overcome these limitations. For example, You can personalize medicine, the combination of the drug, and tune the release of the drug and reduce the number of manufacturing steps. For example, in an article published in 2018, researchers from University of Nottingham used bioprinting for development of tablets with a control release profile. They use paracetamol and as the main drug, and PVP as the biomaterial used for making this combination printable. Then some oval shape, tablets printed. The shape of these tablets, as you can see, is like other tablets, but with different, density. In vitro release, as you can see in the right chart, shows that in release can be controlled with the geometry. As you can see, different geometries lead to different release rate. For example, in the first 50 minutes after release of the drug, 17% of the drug in mesh geometry released, while about 25% of ring shape released and 10% of solid shape released in the same period of time. Also, XRD and FTIR, tests show that the main drug was unaffected during bioprinting process. In last few minutes, I want to talk about our activities in the startup. Our startup, Omida Finance startup, started its work in 2018, and it's founded by some biomedical engineers, students from Sharif University. The team developed the first Iranian bioprinter that you can see here. It's the first prototype in 2019. And this startup tried to cover all the chain of bioprinting value chain, consists of the device, bioprinter, the biomaterial, I mean bioinks, and the final products, I mean bioprinted tissue that can be, transplanted in the human body. Omida Finance sold its first commercial bioprinter to pharmaceutical department of Tehran University in 2019. It was a 2 print heads bioprinter that can print lots of biomaterials and bioink. I will skip this video. Now, from that date until now, Omida Finance developed 4 bioprinters with cutting-edge technologies to meet the wide variety of needs of researchers. As you can see, the feature of these bioprinters are competitive with similar devices from other countries, but of course in a very lower price. Our new approach for development of several modules for bioprinting.
There's 5 minutes left.
Thank you. No problem. For example, we developed lots of modules. For example, low temperature, high temperature, photocuring, FDM, electrospinning, and syringe printers that all of the modules can be used in one devices. This feature enables researchers to access different technologies in one devices. Also, the second part of bioprinting value chain is bioink. We developed about 14 bioinks during last years. Some cell-laden bioinks, for example, soft ink support and gel mob, and some bioinks for printing hard tissue scaffolds. For example, osteoink, apatite bioink based on hydroxyapatite, mineral bioink based on tricalcium phosphate. But our new trend is using decellular extracellular matrix ECM for development of bioink. That algae bone is a bioink based on human bone allografts. There are some videos about printing, application of our bioinks in different methods. For example, in microplastic devices, you can use bioprinting to design and development of microplastic devices very fast in a short time with the capacity of printing cells simultaneously. I will skip these videos very fast. For example, this is a microplastic devices. You can use bioprinters for printing them. But finally, our startup focused on the last part of bioprinting chain, that is printing tissues. The first project is in this part, started a few months ago, is a patient-specific bone scaffold named bone region. The aim of this product is to substitute bone allografts that are now used. With this bone scaffold, there is no concentration, no concern for infection, and so on. This scaffold comes in three shapes: customized mesh, that bone mesh is ideal for maxillofacial surgeries, and bone plugs that is ideal for holes in skull. After successful in vitro and animal tests, we started human tests and clinical trials. The procedure for applying this scaffold for a patient starts with CT images, then 3D model derived from images and used for designing scaffolds. After printing and sterilization and packing, surgeon receive the implant and they use it for the patient. As you can see in the picture, this scaffold absorbs 4 months to 12 months after surgery. This time can be tuned on demand of the surgeon. As you can see, there are some application of these scaffold for real patients, and we hope that we can receive Iranian Food and Drug approval in few months. Our second project in this field is a new project in the field of bioprinting eye cornea. We are using waste of human cornea at hospital to develop a transparent cellular bioink for printing human cornea. And we hope that we finish its prototype in a few months later. We have a bioprinting lab that we developed our bioink and tissues in this lab, and also we provide bioprinting services for researchers in this lab. We have close collaboration with other researchers, especially physicians, for developing tissues using bioprinting techniques. Thank you very much for your time. I hope you enjoy my talk. I want to invite interested people to contact us for receiving more information about devices, bioprinters, bioinks, or tissues. Also, I want to invite kindly professors and researchers for collaboration in desired topics. We are always happy meeting new opportunities and collaborations. Thank you very much.
Thank you, Doctor, for your presentation. It was very informative for us. You can say any question if there's any question. We'll check the chat first.
Actually, I have a question.
Yes, ma'am.
What's your main customers for your products that you develop in Omid Afaryan company?
The main customers for the device, bioprinter, and the bioinks are research institutes and medical universities. But for tissues, for example, bone scaffolds, we work with physicians and patients.
Are you produce them specific for a patient?
Yes, as I said in the presentation, we implant our, patient-specific scaffolds, bone scaffolds into 5 patients until now, and we want to enter the clinical trial to examine it for more patients.
Oh, it's really good. Thank you.
Thank you very much.
There's another question here from the chat. What are the obstacles to be overcome for the full printing of an organ with a bioprinter?
In fact, there are lots of obstacles. For example, to printing a complex organ, you need to print lots of cells simultaneously. And you have a very large, tissue that vascularization.
We can't hear you right now. Is it only me?
Being, problem. I think printing multiple cells simultaneously in a large, tissue is a very complicated process that it's not available until now, but researchers work on it to print lots of, cells in one organ.
Thank you. Thank you.
There's another question.
Thank you. I think Şeyma is having some technical difficulties. Thank you.
She must lose her connection.
Mm-hmm.
Can you hear me?
Yeah, we didn't have any problem hearing your answer. Thank you for your answer, and thank you for your informative presentation.
I was having a connection problem, sorry. It just came. Can you hear me?
Yes, we can hear you now.
Yes.
Okay, did you present Dr. Erciş?
Thank you.
Sundunuz mu Kayhan Hoca'ya hocam?
Şeyma.
Okay.
Macit Hoca'ya teşekkür ediyorduk. Teşekkür edersen sen de.
Okay, thank you, Dr. Majid Hacı Hüseyin Ali, for your speech.
Thank you very much.
Okay. The last topic is bioinformatic algorithms with Dr. Kayhan Erciş, Professor Dr. Kayhan Erciş. He is from our university, Üsküdar University. I will give a brief information about him. He completed his master's degree from Salford University, Electronic Control Engineering. And Professor Erciș's areas of interest are computer networks, graph algorithms, and bioinformatics. He has many publications, and he has published international books on these topics. Now he's currently in Üsküdar University at Computer Engineering Department as a professor. Please, Professor, we are listening to you.
Okay. Hello to everyone. Can you hear me first of all?
Yes, we can.
Yes.
Yes.
Okay. So, if you allow me, I want to share my presentation. Okay. Let's see if we can do it. I can't. Okay, right. And oh. Okay. I don't know. I can't really see my presentation here. All right. One moment, please. Oh God, why can I not see my presentation in here? Okay, I'll try again.
We are ready.
Niye olmuyor ya? Anlayamadım bunu. Şey yapınca, share screen deyince şey gözükmüyor benim presentation.
Hocam aşağılarda olabilir ya da bir tane daha opsiyon var ya orada hani daha fazlası için falan gibi. Show all windows.
Advanced var. Advanced'e bakıyorum. Advanced'te yok. Allah Allah.
İlk önce sunumunuzu açtınız mı hocam masa üstünde?
Evet, açtım onu. Bir daha
Ondan
bakayım.
sonra
Bir dakika. Bir daha bakalım. Problem çıkarttı şimdi. Evet, açık yani bu. Niye olmadı
tamam.
ki?
Buradan share screen tıkladığınızda pencerelerde görmeniz lazım sunumunuzu. Aşağı indirmeden açarsanız direkt gözüküyor hocam.
5 saniye bir daha bakayım. Allah Allah. Şeyi göstermiyor ya. Bir tek sunumu göstermiyor. Onun dışında her şeyi gösteriyordu.
All windows dediniz mi hocam?
Ya ben Linux kullanıyorum.
Onu
Biraz da
bilemiyorum.
farklı. Basic'e baktığımda birkaç tane fazla şey var. Onları yok edeyim. Belki de ondan olabilir. Bir saniye. Bunları yok edeyim. Bir daha bakalım. Pardon. Beklenmedik bir problem bu. Bu değil. Allah Allah, şey yok ya. Sunum yok. Sunum açık halbuki. Başka ne yapabiliriz?
Hocam
böyle bir-
PDF olarak kaydedip PDF olarak açmayı deneseniz.
PDF zaten PDF olarak açtım.
PDF mi?
Yani
Acaba
öyle
dedim PowerPoint'te mi sıkıntı var?
Peki bir saniye, ben bunu bir şekilde başka bir şekilde de açmaya çalışayım. 2 saniyenizi alın. Bu arada yapacağınız başka bir şey varsa yapın. Vakit kaybetmeyin.
Hayır,
Şu
şahsen sizinle.
başka kimse varsa
Yok
benden
hocam
sonra
kalmadı.
evet,
Presentation with you.
sona kalmanın sorunları. Peki bir şekilde başka şekilde açayım. İnşallah olur. Bir tek o hariç hepsini gösteriyor. Evet. Şimdi bir daha deneyelim şansımızı. Açacak gibi. Tamam. 5 saniye. Oldu galiba. Neyse, başka bir programla açtım, o zaman oldu. Şimdi görebiliyor musunuz acaba?
Yes, we can see.
Okay, tamam. Sorry. Okay, now this is a talk about bioinformatics research areas and algorithms. So I'll try not to keep it very long. It's basically a molecular biology review, about 20 minutes. And our research, this is like 15 minutes, and possible future research, that's like 5 minutes. Altogether 40 minutes. I'll try to keep it shorter than this. All right. So what is bioinformatics? It's basically a multidisciplinary topic at the intersection of biology, chemistry, computer science, and statistics. So it requires backgrounds from various disciplines, which we have bioinformatics master program, by the way, and we can have students from very diverse backgrounds. Okay. Now, basically, we can separate bioinformatics research into 2 distinct areas. One is sequence analysis. The other one is network analysis. Now, when we talk about sequence analysis, it's basically DNA, RNA, and protein analysis. Whereas when we talk about network analysis, it's more like the interaction between different biological molecules. Okay, we'll talk about this in more detail. All right. Now, you can hear me and follow the slides properly, yeah? No problems.
Yes hocam.
There's no problem.
All right. Because I'm using a slightly different system, which is Ubuntu of Linux, it can cause problems. All right, life is nice, but there's problems, errors and diseases, okay? Now, these are a couple of things. This is basically a presentation for non-biology people, so it carries a lot of stuff from biology, but I'll just pass quickly because I think everybody here has a lot of background on biology. All right, DNA is basically base pairs, you know, ACGT, okay. Each cell contains full genome, okay. And one very important here is that it doesn't, if you have a longer base pair, it doesn't mean you're more advanced. It's sometimes people may think, okay, if you have a longer chromosome, then you're probably more advanced in evolution-wise, but it's not true. Okay. Now, genes are parts of DNA that carry useful information, as you know. All right, that's like 2% of human DNA. And gene have starting code and stopping code. As you know, this was a very amazing thing when I first learned about it, because it's basically how a computer network works. We have a start code to start the frame and stop. And another amazing thing here, stop codon is 3 different ways. Why? Any ideas? So I thought for a while, because start codon is only one, stop is only, can be 3 different ones, because you want to stop. If you don't stop, you die, okay? So whatever you call it, God, nature, whatever you call it, has taken enough precautions to stop generating new cells, okay? So if you make a mistake and stop codon, if there is a mistake, then it can be tolerated, not the start. Because if you don't start building a protein from a gene, no problem, you don't start the protein. But if you don't stop, it's a big problem, okay? This, I was amazed when I first learned this. All right, these are the codons, as you know, many of you knows this, so I'm just passing by. All right, basic dogma of life, you have from DNA, you have messenger RNA, then the protein, okay? A protein is made of a lot of amino acids, on the average 300, okay? So when we talk about genes, we can talk about the good, the bad, not the ugly, but the silent, okay? The silent ones don't do anything bad. Usually we have the good genes, but many times bad genes, okay? How do they happen? We have deletion of a base pair, or insertion, as here, or substitution. These are all mutations, okay? Now, here comes one of the first very early problems in bioinformatics, which is sequence comparison. This is like a pattern matching problem in computer algorithms. We look at pairwise alignment between different proteins. When these are proteins, each letter is one amino acid, by the way, okay? But a protein is not just a sequence of amino acids, it has a physical shape. Now, they find out that the shape of a protein plays an important role in the function of the protein, okay? So there was this long debate. HIV, big problem for human race, let's say, okay? And they found out that if we can stop folding of HIV virus, then we can stop it. And I think this was the main idea behind manufacturing a drug to protect from HIV, okay? This is called protein folding problem. This is one of the things bioinformatic people may look at, okay? So the sequence of amino acids in a protein is important, but the folding is also important for the function of a protein, okay? So I'm just passing this by. Structure-based drug design, it's a major method for drug discovery. If we can stop folding some bacteria, virus, whatever you call it, we can stop it functioning, which is one basic design method for drugs. Okay, this is HIV protease inhibitor. Okay, so you have something like this in the middle, which can stop folding. Right, this is a gene regulation network. It's one of the simple networks we want to have a look. You have the gene, messenger RNA, then the protein. This is the usual sequence, okay? But there are possibilities that the protein produced may actually influence the production of messenger RNA in another gene, all right? Okay, that's called a GRN. It's a network in terms of computer terminology. So we can just follow what's going on in this network, all right? And gene expression is just another thing you probably know. We all have the same genes in every cell, but we have cell for eyes, cell for legs, etc. They all function differently. Okay. So what are the problems for bioinformatics people? Number one problem, gene finding. How do we actually locate genes? All right. This is a computationally very heavily computation problem. Why? Because we search for very small pieces of useful information in a very long, large data set. It is extremely difficult, okay? How do they do it? Well, you may think a gene has a starting codon and a stop codon. So why is it so difficult? But the search space is so large, then people usually do some statistical methods. They compare similar species, and if they find the same kind of structure along the same places, okay, that's a good location to search for a gene. Okay, that's number one problem. The second one is sequence alignment. All right, you assume that this is the first line is one species, the other one, second one is another one, and we try to find out how they are related. Are they close enough to each other? Okay, if you find they're quite close to each other, then we can say that they come from the same origin. Okay, there are lots of algorithms for this. It's like the beginning of bioinformatics, really. People just started with alignment algorithms, as far as I know. Okay. Another big problem in bioinformatics, tandem repeats, meaning some code where you see in green here is repeated. All right. So this is again a lot of A's and G's, a lot of A's and G's. These are all repeated many times. Okay. Most of the time, people thought this can happen arbitrarily, but it does not, because they find out large tandem repeats in Alzheimer's. Huntington's carrier is a very, I don't know whether you've heard about it. It's another neurological disorder. Some cancer types, we observe a lot of tandem repeats. So the problem is this: if we can discover big tandem repeats in DNA, okay, we can say that that person may have some kind of problem in terms of, Alzheimer's, Huntington's, or some cancer types for early diagnosis, okay? Yet another problem, genome rearrangement. Now, let's say you have some kind of genes in order. In another organism, same genes with different order can cause a totally different organism. Here you have cabbage and turnip. Okay, they don't look like at all, but they exactly have the same genes in different order. Okay, this is slide showing. So this is turnip 1, 2, 3, 4, 5. In cabbage, it's 1, -5, 4, -3, 2. -5 means the genes nucleotide order is reversed too. So we can say that sometime in the past, turnip chromosomes broke down and pasted differently somehow. There was some kind of
reversing of the gene order, and we have a totally different species, organism. Okay, it does happen. The chromosome breaks up at some point, and it's glued back as inverted. This is a known problem. Okay, well, it does happen for goodness sometimes. Okay, but finding the order, for example, can we just deduce that cabbage and turnip come from the same organism? This is another bioinformatics problem. I have a student working on this, by the way. Okay, now phylogenetics is a tree of life. All right, now we have DNA information on living people in the world, living species, living organisms in the world. Can we deduce, can we find out some kind of relationship between these in terms of ancestry relations, who comes from who, etc.? This does help to find, for example, mutation of viruses, even in our case, this infamous C19 virus. Okay, if we can find out, if we can work out the phylogenetics relation, again, this is an algorithmic problem. All right, we only have the current data. We don't know previous ancestors' data normally. Sometimes we do, because whenever we do some kind of search in like old mummies, this and that, we can find some DNA, but typically, commonly, we would have DNA information for the current population. And can we deduce some kind of relationship between those people in terms of who is the ancestor of who? Okay. All right, one nice problem here. Human migration out of Africa. This is the basic thesis that how human actually moved from Africa to Europe and various parts of the world. This is an accepted thesis. Out of Africa, it's called. Okay, probably know it. And I find out in two different locations, one in Germany, one in Caucasus, they found two Neanderthal species, okay? And they checked. Now, this is very interesting. Europeans have 3% on the average Neanderthal admixture, okay? Africans do not have any Neanderthal admixture, which tells us Africans are truly Homo sapiens. What a blow to some philosophers and even scientists who claim that Africa, okay, they're inferior, this and that. What a terrible blow to those guys. Science is nice. All right. Now, HIV transmission. This is one of the first cases in the world that phylogenetic investigation turned out to be very, very useful to find out that to resolve a case in the United States. This woman claimed that she received HIV virus from her dentist. Dentist said no. She was my ex-lover, she's just making it up, this and that. And in the end, they find out, okay, just by examining the HIV from the dentist and the woman, they find out phylogenetically, they come from the same origin. This was the first case. Phylogenetic analysis was used in the court case. Okay, so phylogenetics analysis is useful for other purposes. All right, now the second part is this was all about sequences, all right? A sequence is basically like a stream of symbols, ACGT or some amino acid symbols. But when we're talking about networks, we're talking about some kind of nodes which are connected to each other, okay? So we have PPI networks, one big research area, protein interaction networks in the cell. On the average, there's like 50,000 or 60,000, between 50 and 60,000 proteins in a cell.
they are connected to each other by using signals. Okay, so this is one very important area. This is where we work, by the way. This is my basic research area is PPI networks. Okay, we've not really looked at metabolic networks and pathways. These are more complicated, really. Gene regulation networks is just another one, and brain functional networks, which is very suitable for Üsküdar University, I believe. Okay, this is another biological network. Okay, this is PPI network of yeast. Yeast is considered as an example for many protein networks because it's not so complicated. Okay, now when you look at a network like this, you find out it is not random. It's a strange network. You have a lot of nodes, but only some are hubs. Hub means a lot of connections. Okay, now from one end to the other end of the network, which is called the diameter of the network, is comparatively very small. When you consider the size of the network, there's like 20, 30,000 proteins here, maybe, okay? And from one end to the other end, you can actually reach like 8, 10 hubs. These are called small world networks. This was a famous experiment by Milgram. There's a film about it, by the way. It's called The Experimenter. Stanley Milgram, very famous sociologist and psychologist. He said everyone is connected to another person in the world in 6 hubs. It turns out protein networks are small world networks, and so is internet. Very fascinating stuff. And brain functional network. All right, you put probes in brain, and this is not a statistical network. It's a dynamic network. So you give some kind of stimuli to the patient and work out the connections. It's called BFN. All right. So I have a student working in this area and trying to analyze brain functional networks. Okay. So it's a complex network. A complex network is a small world network with scale free, we call it, because as you can see from this example, there are just few high degree nodes, hubs, but most of the nodes in this network are very scarcely collected to others. But there's a couple of them, several of them have very large connections. Now, these proteins, if they fail, they may be the cause of really serious diseases like cancer. Okay, so if a small node fails, for example, let's do a rewind. If there's a mutation in the gene for a small protein, okay, possibly this can be tolerated. But if there's a mutation in the gene that translates to a protein, something like this. I don't know whether you can see the hand, small hand in here, but you know, this is the one. Anyway, let me take it here. Just below the line, you have a hub here. If there's a problem, if there's a mutation in the gene that produces this protein, that probably will cause cancer. Okay, so what we're saying here in a sense is that analyzing a network gives us a lot of information about the wellness or the disease state of an organism. Okay, so there's a couple of brain functional network diagrams here. I'm just passing by. Well, this is one book I had authored a while ago. It's called Complex Networks, by the way. It has a lot of information on biological networks, basically, and internet. It turns out Facebook or the protein interaction network in a cell or the internet are no different, which is incredible. People never thought about this. When they found out they're all the same, well, it was just incredible. Okay. So what do we do in biological networks? We do, this is like the second part on how we deal with this stuff. We deal with the stuff like analysis. We do network motive search, alignment. I'll come to that, okay? We need efficient algorithms to start off. What we do basically as computer scientists is algorithms is the key to our research, okay? Now, we find out, even for small numbers of n, if your algorithm works like 2 to the n times, like even for 100, it takes, for example, 2 to the n is 10 to the 17 years. Well, there will be no sun by that time, so they say. No Milky Way, no nothing, okay? So 100 is a very modest number. I'm talking about 50,000, 60,000 proteins. So we're lost, okay? To solve bioinformatics problems, data is so large. Our algorithms, we just have to design very efficient algorithms, which is the core topic in bioinformatics. In order to solve a biological problem, we have to design very nice, proper algorithms, which is extremely difficult. Sometimes we don't have proper algorithms. What we do is we use approximation algorithms, which are algorithms that do not find the optimal solutions, but they find suboptimal solutions. Most of the time, even this is not possible. We use heuristics, which are common sense rules. We show them, they work experimentally for most of the inputs, okay? All right, one big problem for us, network motifs. Whenever you look at some kind of biological network, you find a common pattern that repeats itself, okay? Now, this is like looking at a Lego vehicle and you find out some small red piece is repeating itself. You have many red small stuff and you wonder, okay, this must have some kind of reason for it, all right? And you find out if there's a motive, there must be some kind of significance attached to it. Some of the common motifs we find, these are the very simple ones, feed-forward loop, okay? So you have a node in here, it sends a signal to this one, and this one sends to the other one. Again, we see some kind of fault tolerance at the bottom of nature, okay? So this guy is just making sure that data is sent to this one, all right? Now, brain networks have some kind of motifs. Now, we may think, all right, if we find a motif in a brain functional network, it must mean there is some kind of significance attached to it. Maybe it's a low-level intelligence building block. Okay, this is a very hot research area which we're working, okay, with one student. But it's a great challenge, obviously. Okay, so some information about your brain and brain functional network of an anesthetized cat. Okay, now here comes a very important topic. Brain functional network of a healthy person on the left and the person with schizophrenia. You can see the difference easily, all right? This one is like a more connected network. This one, you can see a lot of different nodes. All right, now, in our study, what we're doing is, if we can identify these characteristics of persons with schizophrenia or Alzheimer's disease, then we can come up with a general structure for these people. Eventually, we can build a simple equipment, okay? You go to doctor and doctor takes all your BFN EEGs and says, okay, 80% you have Alzheimer's disease. It's a big challenge, it's a revolutionary stuff. I think, well, it can be done. We haven't done it, but it can be done, okay? Also, if you know that some connections are missing, maybe there's a possibility to repair those connections to bring that person back to normal life, to healthy condition. Okay, it's a grand challenge. I can't say we're just even doing like the first couple of steps. It's a big challenge. Okay. There's also structural brain networks, which is basically networks which don't change with time, but BFNs is more important. Yeah, I think I'm coming towards the end. Analysis of BFNs may provide insight to the mechanisms of diseases, but all of these depends on graph theoretical analysis. Okay. Now, there's a lot of studies on BFNs. Okay. Actually, there was this professor, assistant professor. She worked on BFN data here, but she's not here at Üsküdar University anymore. So we got her data, but we had some problem in analyzing that data, unfortunately. Okay, right. So there's a lot of stuff on. So we come to what we do as our research. Okay, number one, we did a parallel genome reversal algorithm. This was like 5-6 years ago. And we provided parallel approximation algorithms to find out, you have different type of organisms. We find out if some kind of genes are reversed in the other one. Okay. Phylogenetic tree construction, this one, another master thesis, which I did with a master student. This dates back even more. I think it's like 8-9 years, maybe 10 years. I can't remember. What we did was you can actually specify people based on their Y DNA haplogroups. So there's like 28 haplogroups. Okay, we took G haplogroup. We calculated closeness values and we used clustering first, then neighbor joining algorithm to build trees, then merged again. It's a long, tedious, difficult process. In the end, we built phylogenetic trees like this. It was published in a very good journal. Okay, this was a while ago. We used data from GM406 haplogroup, which is the group I belong, by the way. Why, you may ask why? Because I could access this data. Okay. And this is a very, very funny story. Maybe I should say this. Okay, there was this guy, an Italian millionaire, is from Canada, Italian origin. Okay, so he used to send us some money. He used to send money to me, so we would help refugees in Turkey, elsewhere. Okay, so he's a very, very rich man, owns mines, this and that in Canada. Frank Ustra, you can just Google him. All right. Now, one day I find out, just searching through my DNA, RGS and Ustra, we're very close with him, DNA-wise, which was incredible. Same haplogroup and values are very close. Okay, so I asked him, Frank, I am very close to a Justra, and probably there's so many Justras in the world. His answer was, no, there's only a few Justras. We are related. That was very funny. Okay, now we did also parallel network motif search. This is a more recent research. Again, research for motifs in yeast or other organisms. E. coli. We looked at two organisms. One is yeast, the other one is E. coli, because they are simple. Okay, and we did a lot of. This was again a very tough thesis, PhD thesis. She just finished about a year ago. This was at EGE, and we came up with better running times than current literature. It was in a good publication, very recent, a few months ago. Okay, this was our results. And the current ongoing project is network alignment. We try to find out how one network resembles to another one. As in the schizophrenia or Alzheimer's disease case, if we can find some people who have similar network structures, then we can say, okay, these Alzheimer people we can characterize them with a Bayes network. And if somebody comes to the doctor with some diagnosis, okay, the doctor simply can use an algorithm like ours, whether that person's network is similar to the Bayes network. If it's similar, okay, you can say, all right, 75% you can be Alzheimer. That's much easier than going through a lot of tests, details, this and that, okay? And also, it's one step towards building, modifying, curing that disease. Okay, but network alignment, there's a lot of algorithms for it. Strange enough, some Turkish people, not in Turkey, these are guys in United States and Canada, have really taken a good look at this problem. One is Koyutürk, the other one is Erten Alada. Okay, Mavish, probably he had a probably daughter with blue eyes, I believe. Why would he put a name like this otherwise? Okay, these are the stuff. And there's a lot of research going on in network alignment too. Okay, this is another book I authored. This is more recent than the other one. This is basically just on bioinformatics. We combine sequence analysis with network analysis. It's the first book that does this. Typically, you would have bioinformatics books on just sequence analysis, but this is network and sequence analysis, so it was the first one. It did not sell much. There's only a few people interested in this area. Okay, so we go on working with my students on areas like protein networks, brain functional networks. Here, I did not really move on to proteomics or biological pathways. This is like, it requires more biological information, which I do not have. But network structures of biological networks is what we do. So I basically use Python or other languages to do network analysis. And basically the core of our research is building algorithms. Nice, good algorithms, parallel algorithms. This is our basic research. I'm not a biologist, but I just would like to have the biological problem as a model, as a graph of some kind, and we try to solve it. Okay, these are a couple of things. That's all I want to say. Thank you. If you have any questions, I'll be pleased to answer.
Thank you, Doctor. Is there any question? Dear friends, dear participants, you can write on the chat board. Doctor, I have a question. It's echo, making echo. May I ask, Doctor?
Yeah, sure.
Which biological network is the most complex for you?
Which biological networks?
The most complex for you when you observe it.
Okay, there are two sides of this question. It's a nice question. There are two sides. If the number of nodes in the network is important, then protein networks, they're very crowded. They're large networks, but they are very much investigated. Okay, there's a lot of research on them. But in terms of understanding concepts, brain functional networks, they are very, very little investigated, but they are smaller networks. I don't know, because people don't have this data from EEG, I believe, until recent times, proper data. Now they can build brain functional networks. There's a lot of databases all around the world. You just have to go for brain network connectome data. It's called connectome. So you can just find, for example, UCLA has a large database for people with...
for brain function, right? Connectome.
Sorry?
Sorry. This database, sorry, for the brain function, right?
Yeah.
Connectome, you said.
Yeah. UCLA has such one for healthy people and people with schizophrenia and Alzheimer's, etc. So, you can just download data from there, compare data. It requires algorithmic approaches and some mathematics, some statistical analysis, but they're not so difficult. Basically, my general conclusion is that bioinformatics, why hasn't gone so fast? Because you need people with background in algorithms, some biology, some network, some programming. You know, you need people who have to know a couple stuff at the same time.
Thank you, Doctor,
Okay.
for your ideas and explanation. Is there any question, by the way? Yes, Ömer Şamdin has a question. Yes, Ömer, we are listening to you. Please ask.
Okay, I can't hear anyone, by the way.
We're waiting the question, Doctor.
He's gonna write it to the chat board, hocam.
Now, how am I going to see the chat board? It's disappeared.
No problem,
A
Şeyma.
long question, I guess.
Probably.
Shema, can you read it, please? Because I've lost my chat board. I don't know what's going on.
The question is long, I think. We are waiting.
I will read.
Yeah,
Yes.
okay. We have a bioinformatics master program at Üsküdar University, by the way, just opened. No students yet.
For protein folding, is there any specific factors that specify how the protein will fold?
I don't exactly know. I'm not so much informed in this topic, but all I know is the protein functions based on its folding. What it's basically, I would think the sequence of amino acids determine how it's gonna fold. I would think, but I'm not sure how this is biologically initiated. There must be some kind of mechanism for it. But if we can stop it, you can stop the disease. Basically, that is known.
I wish we could ask this question to Hasan Demirci. Maybe can reply. Thank
He's not
you.
here.
Okay.
Is there any question? Another question? Okay.
Okay.
Thank you, Doctor, for joining us,
Thank you.
for sharing your experience and research. Okay, so today we completed our session. Thank you for joining us, dear participants, and thank you for honoring us, dear doctors, dear professors. I hope we can organize next week. We hope this. We hope you enjoyed it and you found it very informative, very beneficial. If you have any question, if you have any problem, you can send email to me as a vice chair of club. I will share my email, and you can send a mail from to Mehmet Akyıldız. He's the chair of club. We will share our emails on the chat part. Thank you, all of you. Yes,
Thank you
it was
all for your
Thank you, hocam.
participation.
Thank you
Let
so
me
much. Thank you, everyone, for listening. It was a nice event. We hope to see you next year.
Your certificate will be appearing, and we will send them as soon as possible, but we will compare them with a registration form and the Zoom list. We will see who will attend and how much time attend. So we will compare them, then we will send you as soon as possible. Thank you, all of you.
Thank you, everybody. Bye-bye.